in the quiet imagination of deep time, long before human memory, long before forests, mountains, or even the simplest familiar creatures, the Earth existed in a state almost unrecognizable to us now. It was a young world, still cooling from its violent beginnings, its surface shaped by slow oceans of molten rock and unsettled skies that carried more steam and volcanic gases than true weather. Yet even in this ancient stillness, there was a sense of gradual change, of a planet preparing itself for the gentle unfolding of life that would one day transform every corner of its surface. In this part of our lecture, we linger within that early moment, letting the mind drift over the landscapes of a world not yet alive, a world quiet enough to imagine the faint stirrings of life's first expressions. During Earth's earliest ages, the oceans were only beginning to form. As the planet cooled, water vapor condensed into vast clouds that circled the globe, releasing rain that fell for thousands upon thousands of years, pooling into basins and carving shallow shapes into the cooled crust. These first oceans were warm, mineral-rich, and dimly lit beneath a sky heavy with volcanic ash. Sunlight reached the surface, but filtered and softened, as though seen through layers of fog. And in the warm shallows of these oceans, chemistry was shifting, organizing itself from simple arrangements into more complex forms. Guided only by the gentle laws of nature, molecules collided, connected, broke apart, and reformed, each reaction opening the possibility for something slightly more intricate. It is here, in these quiet tides, that life's story begins. Life does not appear all at once. It emerges softly, through processes we still strive to understand. According to what is broadly accepted in biology, every living organism is composed of cells, and all cells arise from pre-existing cells, a principle known as the cell theory. Yet, in these earliest moments, before cells truly existed, there were only the ingredients that would later form them. Carbon, introduced partly through volcanic eruptions, combined with elements like hydrogen, oxygen, and nitrogen to form amino acids, the small, essential building blocks of proteins. These building blocks drifted through the water in enormous abundance, interacting through slow chemical rhythms made possible only in oceans where temperature, energy, and time met in the right ways. As these molecules accumulated, new structures gradually formed. Some became long chains, capable of holding simple information. Others folded into compact shapes that performed basic tasks, eventually giving rise to protoenzymes, molecules that could speed up reactions around them. Still others arranged themselves into membranes, thin but stable, capable of separating an inside from an outside. These were not yet living cells, but they were precursors to life, the first hints of organization in a world otherwise ruled entirely by geology and chemistry. And through calm repetition, over vast expanses of time, these structures slowly became more refined, more consistent, and more capable of cooperating with one another. The early Earth was not peaceful in a familiar sense, but its changes, though dramatic on geological scales, were gradual enough to allow these molecular systems to persist. Cooling continued at a pace that permitted water to stay liquid. Volcanic activity, while frequent, lightning storms and ultraviolet light provided steady energy for chemical transformations. Through this interplay of conditions, simple molecules eventually evolved into more complex ones, giving rise to entities capable of storing, using, and passing along information. Among these, RNA-like molecules are thought to have been especially significant. Evidence suggests that such molecules were capable not only of carrying information but also of catalyzing the reactions needed to replicate themselves. In many ways, these early self-replicating structures represent one of the first major thresholds in the emergence of life. Even at this point, the Earth remained largely still and silent by our standards. There were no plants to sway in the wind, no animals to swim through the seas. The atmosphere was dominated by carbon dioxide, methane, ammonia, and other gases. Yet in this quiet, nutrient-rich environment, early living systems could slowly arise and stabilize. Life did not require peace or comfort. It required consistency, patience, and opportunity. And in the warm oceans of early Earth, those conditions were quietly present. Once the first true cells appeared small, simple, and without nuclei, they marked a profound transformation in Earth's history. These were the prokaryotes, ancestors of bacteria and archaea, organisms composed of only a single cell, yet capable of performing all the basic functions of life. They inhaled and exhaled in their own microscopic way, converted nutrients into energy, expelled waste, and reproduced through simple division. Despite their simplicity, these early cells carried within them the seeds of all future complexity, from the tissues of plants and animals to the intricate structures of fungi and algae. Over time, as these cells divided and spread across the planet's oceans, they began to shape their surroundings, altering the chemistry of the water and atmosphere very gradually. Their presence marked the beginning of a new interconnection between life and environment, one that would continue deep into the present. Still, at this early stage, the oceans remained the sole cradle of life. The land was barren, quiet, and untouched, with no soils, no roots, and no sounds beyond the shifting of wind and stone. In the water, life existed mostly as drifting shapes so small they were invisible to the naked eye. Under the surface, sunlight wavered gently, filtered through minerals and gases, creating a world of soft luminosity where these tiny cells carried out their slow metabolic work. The rhythm of the early ocean became the rhythm of life itself, a steady pulse of reactions, divisions, and gradual adaptations. As millions of years passed, Earth's surface continued to stabilize. The planet's orbit and tilt settled into patterns that allowed more predictable climates. The oceans grew deeper and more interconnected. Gradually, the conditions that supported early cells became even more favorable, allowing them to spread widely and thrive in many different environments within the sea. Some cells preferred warm shallows near geothermal vents, while others drifted into cooler open waters. Each environment offered different challenges and opportunities, guiding the long path of evolutionary change through calm, steady processes. Biology reminds us that life emerges not in leaps but in subtle shifts, through variation and adaptation, accumulating step by step, intricacy arising from simplicity across incomprehensible stretches of time. And so, in these earliest days, Earth transformed quietly from a lifeless sphere of rock and water into a world where the first living systems flickered into being. These simple cells did not yet form multicellular organisms. They did not yet communicate or build tissues or coordinate their efforts. But they represented life's first breath, the beginning of a story that would eventually lead to multicellular forms, complex structures, and the rich diversity of living beings we see today. For now, though, the world remained gentle and uncomplicated, shaped by tides, sunlight, and chemistry, all working together in patient harmony. In this calm environment, we find the foundation of everything to come. The origins of multicellular life lie not in sudden change, but in this long. It is a story that begins not with noise or conflict, but with water, warmth, and time elements that nurtured the earliest expressions of life and welcomed them into existence. As we leave this opening part of our exploration, we carry with us the sense of a world just beginning to awaken, its oceans filled with the quiet potential that would one day unfold into the vast tapestry of multicellular life. 2. The nature of life's smallest units. To understand the origins of multicellular life, it is helpful to rest for a moment with the simplest building blocks of biology, letting the mind settle into the gentle rhythm of what a cell is and why it matters. A cell may seem small, almost impossibly small, yet it contains within its delicate boundaries the entire machinery required for life. Before life could ever become multicellular, before tissues, organs, or organisms could emerge, cells had to exist, and they had to do so in a world that was still smoothing its rough edges. By exploring the nature of cells, we also come to understand the quiet foundation that made all later complexity possible. It is as though the early Earth crafted its first alphabet, and from those characters, the long narrative of evolution could slowly begin its work. A cell is far more than a tiny droplet of material drifting through ancient seas. It is the basic unit of life, containing the essential structures and processes that allow living things to breathe, to eat, to grow, to respond, and to reproduce. Even the smallest cell, invisible to the naked eye, is an intricate, self-sustaining world unto itself. It is capable of taking in nutrients from its environment, converting those nutrients into usable energy, maintaining stable internal conditions, and dividing to form new cells. According to the cell theory, all living organisms are composed of cells, and all new cells arise from the division of pre-existing cells. This principle anchors our understanding of how life grows and persists through time, and it shapes every branch of biology, from evolutionary theory to modern genetics. When early Earth provided the right conditions for life to begin warm oceans, abundant minerals, and favorable chemical energy, the earliest living cells appeared, simple but complete. These first cells were prokaryotic, organisms without a nucleus and without the elaborate internal structures seen in more complex cells. Bacteria and archaea belong to this ancient lineage, and their forms have persisted across billions of years with remarkable stability. They are among the earliest organisms to have appeared on Earth, thriving in marine waters, freshwater environments, and even in extreme conditions such as salty pools, oxygen-poor swamps, volcanic springs, and geothermal vents. Their adaptive range suggests that from the very beginning, life was already capable of finding ways to survive in a world that offered both opportunity and challenge. These early cells were incredibly small, simpler than the cells found in plants and animals today, but they carried out all the essential functions that define life. They had membranes, thin and flexible, that enclosed their internal contents and protected them from the surrounding environment. They contained molecules of DNA, holding the information needed for reproduction and the building of proteins. They housed enzymes capable of guiding chemical reactions, allowing them to extract energy from nutrients. And each cell had the ability to sense changes in its environment, responding gently to variations in light, heat, chemicals, and other influences. The world of a cell may be quiet, but it is never still. Inside, countless reactions unfold at every moment, sustaining the delicate balance of life and maintaining the continuity that allows evolution to proceed. As evolution continued, another major shift occurred in the story of cellular life. Some cells grew more complex, forming internal compartments surrounded by membranes. These were the first eukaryotic cells, the ancestors of plants, animals, fungi, and protists. Unlike prokaryotic cells, eukaryotes contain a nucleus where DNA is safely enclosed, as well as specialized organelles like mitochondria and, in plants, chloroplasts. These internal structures gave eukaryotic cells new capacities, greater efficiency, better organization, and the ability to handle more intricate tasks. Their development marks one of the most important transitions in the history of life. The presence of these organelles suggests that early cells may have formed partnerships with one another, with smaller cells living inside larger ones in mutually beneficial arrangements. Over immense spans of time, these partnerships became permanent, and the result was a cell type capable of supporting the complexity required for multicellular organisms. With the emergence of eukaryotes, cells were no longer limited to simple tasks. They could begin to divide labor among their internal components, allowing different parts of the cell to specialize in different kinds of work. This internal specialization mirrors, in a quiet and early way, the later division of labor seen in multicellular organisms, where entire groups of cells take on unique roles. In this sense, eukaryotic cells represent a small echo of the organizational principles that would one day make animals, plants, and fungi possible. Before multicellularity could arise, cells themselves had to become capable of the internal coordination required for larger systems. Despite their variety, all cells share certain characteristics. They are highly organized, containing many molecules arranged in precise patterns. They respond to external stimuli, adjusting their behavior when conditions change. They grow, reproduce, and carry genes that pass hereditary information from one generation to the next. And they maintain homeostasis, keeping their internal environment stable despite changes happening outside. These traits create a sense of continuity that stretches across the tree of life, binding all organisms together. While evolution has produced extraordinary diversity, the underlying principles that govern life remain consistent across species, connecting the smallest bacteria to the largest animals. Life's diversity emerges through variation in genes, through changes in DNA introduced by mutation and recombination. These variations shape the traits of each organism, influencing everything from cell shape to metabolic patterns. As cells divide and populations grow, these traits can spread or diminish depending on environmental pressures. In this way, even before multicellular organisms existed, evolution was already sculpting the living world, guiding populations of cells in subtle, gradual directions. These earliest evolutionary steps created the conditions necessary for multicellularity to emerge much later. The building blocks were being refined through patient repetition, cells becoming slightly more efficient, slightly more stable, slightly more capable of interacting with one another. While individual cells are remarkable on their own, their true potential emerges when they are considered as part of larger systems. Even in the earliest phases of evolution, cells began forming associations with one another. Some divided but did not fully separate, remaining loosely connected as clusters. Others communicated with chemical signals. These early collaborations may have been the first hints of multicellular behavior, arising long before multicellular organisms truly existed. Yet each small change contributed to the larger narrative, building a foundation from which multicellularity could eventually grow. Though cells vary widely in size, shape, and complexity, every cell reflects the same elegant principle. Life is organized, purposeful, and continuous. Nothing about a cell is accidental. Its membranes, molecules, and internal structures work together in harmony, maintaining balance through countless quiet processes. This inner harmony allowed cells not only to exist, but to persist, surviving through shifts in environment, climate, and geological change. By the time multicellular organisms appeared, cells had already mastered the art of stability. They had already demonstrated the ability to cooperate, adapt, and respond to their surroundings. These qualities made multicellularity not only possible but inevitable over long stretches of evolutionary time. As we prepare to step gently into the next part of this lecture, we carry with us a sense of the quiet elegance and extraordinary resilience of life's smallest units. Cells are the foundation upon which every stage of evolution is built, and their story spanning billions of years is the story from which multicellularity would later rise. By understanding the nature of cells, we are better able to appreciate the long, peaceful unfolding of life from its humble beginnings toward the rich, interconnected world we know today. 3 prokaryotes, the first wanderers of the sea, long before the world knew plants or animals, before any creature swam, crawled, or grew roots into the soil, life existed in a form so small and so simple that it could drift unnoticed through even the calmest ancient waters. These earliest beings were the prokaryotes, tiny single-celled organisms without nuclei or complex internal structures. Though their bodies were little more than delicate membranes surrounding fluid and molecules, they carried within them the spark of life that would one day unfold into all biological diversity. To enter their world is to step into a time when Earth's oceans were warm, mineral-rich, and profoundly quiet. A stillness broken only by the ceaseless drift of microscopic forms learning, in their own slow way, how to survive. Prokaryotes emerged at a time when Earth was dramatically different from what we experience now. The atmosphere lacked oxygen, consisting instead of gases released from volcanic activity: methane, ammonia, carbon dioxide, nitrogen, and water vapor. Sunlight reached the surface through this hazy air, illuminating seas that were young, warm, and filled with dissolved minerals. Into this world, prokaryotic cells appeared bacteria and archaea, representing the earliest known living organisms. They were incredibly small, often only a few micrometers across, yet they were fully alive, capable of performing every function needed to sustain themselves, taking in nutrients, producing energy, removing waste, and reproducing. These cells, simple as they were, marked one of the most important transitions in the history of the planet. Their simplicity did not limit their resilience. Prokaryotes thrived in environments that might seem hostile to modern forms of life. They colonized land, water, and even the most extreme habitats: acidic pools, deep-sea vents fed by volcanic heat, salty lakes, and stagnant swamps without oxygen. Some archaea adapted to environments so hot, cold, or chemically intense that no other form of life could survive there. Their success in such diverse places suggests that from the beginning, life on Earth possessed a remarkable adaptability, a quiet determination to persist no matter the circumstance. The earliest oceans were not gentle in the way we imagine calm seas today. They fluctuated in temperature, chemistry, and depth over long periods. Yet prokaryotes spread through them with patient confidence, finding niches that allowed them to continue dividing and evolving. Though they lacked nuclei, prokaryotic cells contain DNA, a molecule capable of storing the essential instructions governing their growth and reproduction. This DNA floated in the cytoplasm, unconfined by compartments, yet fully functional as the keeper of hereditary information. As these cells divided, the DNA was copied and passed to daughter cells, preserving genetic continuity. This simple form of replication provided the earliest path through which traits could be inherited, laying the foundation for evolution through natural selection. Over time, random mutations introduced subtle differences into the DNA. Some variations provided advantages under certain conditions, while others faded quietly as cells failed to thrive. These slow patterns of change shaped the earliest lineages of life, guiding them toward greater diversity and resilience. Even in their simplicity, prokaryotes displayed remarkable versatility. They adopted a range of shapes, spherical, rod-like, and spiral forms that allowed them to move, divide, and interact with their environment in ways suited to their needs. Some drifted freely on ocean currents, while others attached firmly to mineral surfaces or formed thin layers called biofilms. These biofilms, composed of countless cells living closely together, represented some of the earliest examples of cooperative behavior long before multicellularity emerged. Within these layers, prokaryotes shared nutrients, communicated through chemical signals, and protected one another from environmental stress. Though each cell lived independently, there was a quiet sense of community, a soft foreshadowing of the more sophisticated collaborations that would evolve much later. One of the most important early innovations in prokaryotic life was the ability to extract energy from the environment. Some prokaryotes relied on chemical reactions involving sulfur, iron, or hydrogen processes that required no sunlight. These organisms thrived near geothermal vents and volcanic springs, where heat and minerals provided the fuel for their metabolism. Others developed the ability to harness sunlight through primitive forms of photosynthesis. These early photosynthetic cells altered the course of Earth's history by releasing oxygen as a byproduct. Though this shift occurred slowly, The accumulation of oxygen in the atmosphere, known as the Great Oxygenation Event, transformed Earth's chemistry and paved the way for more complex forms of life. The oceans gradually changed as well, becoming environments where new metabolic strategies could take hold. Oxygen brought both opportunity and challenge, enabling efficient energy production while also creating the need for structures that could manage reactive molecules. As oxygen levels increased, some prokaryotes adapted to this new element, developing mechanisms to use it in their metabolism. Others retreated into oxygen-poor environments where they could continue living as they always had. This divergence marked an early branching of life, with different groups finding different solutions to the same environmental pressures. Through these changes, prokaryotes demonstrated a capacity for evolution that would later support the emergence of eukaryotic cells and, eventually, multicellular organisms. Their long history of adaptation reflects the quiet persistence required for life to take root and flourish across billions of years. Despite their small size, prokaryotes played a profound role in shaping the planet. They influenced the chemistry of oceans, contributing to nutrient cycles that affect every layer of the marine environment. They interacted with rocks and minerals, altering the geology of the seafloor over long periods. Some prokaryotes even formed structures known as stromatolites, layered mounds, created by trapped sediment and microbial growth. These formations, found in ancient rock, provide some of the earliest fossil evidence of life on Earth. They stand as calm reminders of a world once ruled entirely by microscopic communities. Prokaryotes also introduced the first forms of ecological interaction. They competed for resources, cooperated in shared environments, and influenced one another's success through simple yet meaningful relationships. These interactions created the earliest ecosystems, quiet, stable networks of life built entirely from single-celled organisms. Even without multicellularity, the world was already complex in its own subtle way, shaped by countless invisible processes unfolding in oceans and shallow pools. As we look back on the era dominated by prokaryotes, we see a world that was vast in time, yet modest in form. Life had not yet taken on the shapes we recognize, but the foundations of biology were already firmly in place. Cells existed, reproduced, and diversified. Energy flowed through delicate chemical pathways. Genes passed from one generation to the next. Natural selection sculpted populations, guiding them towards stability and resilience. The first wanderers of the sea may have been small, but they carried within them the beginnings of everything to come. Without prokaryotes, there would be no eukaryotic cells, no multicellular organisms, no plants, animals, or fungi. Their story is the opening chapter of evolution, the moment when life learned to exist, endure, and adapt. As we leave this part of our exploration, we carry with us an appreciation for the humble origins of life, where the simplest cells drifted through ancient waters, unaware that they were shaping the future of the planet. 4. The gift of cooperation. In the long, unhurried history of life on Earth, one of the most profound transitions did not arrive in a single moment, nor did it emerge through dramatic changes. Instead, it unfolded gently, almost imperceptibly, as single cells discovered the simple advantage of being near one another. Before the world could host multicellular organisms, before tissues or organs could arise, there had to be a quieter, earlier step. The realization, shaped by nature's patient hand, that survival could be eased through cooperation. This cooperation did not begin with intention or awareness. It began as soft patterns of sticking together, lingering after division, or drifting into shared spaces where nutrients collected. Over time, these small moments of closeness allowed life to explore new possibilities, creating the earliest hints of collective existence among organisms that had once lived entirely alone. In the earliest seas, prokaryotic cells divided as they always had, splitting their contents in two and drifting away into the surrounding water. Yet sometimes, due to the nature of their membranes or the viscosity of the ancient ocean, daughter cells remained close together for a little longer than usual. Perhaps they clung lightly to one another, held by soft chemical bonds or the remnants of the cell wall that had not yet fully separated. This seemingly insignificant closeness allowed them to share resources more effectively. For example, metabolic byproducts produced by one cell might become the nutrients that another cell could use. By being near each other, cells inadvertently created tiny pockets of stability, small groups that weathered shifts in temperature, salinity, or chemical conditions better than isolated individuals. In these early associations, no cell controlled another. There was no communication yet, no coordination, no collective purpose, but the environment rewarded the cells that remained close in subtle ways. When nutrients were scarce, clusters of cells could make better use of the limited resources available. When environmental conditions changed, groups offered more microhabitats than a single cell could ever create. These early benefits formed the quiet groundwork for cooperation. Though primitive, this cooperation was one of the first steps away from complete individuality, hinting at the evolutionary path that would eventually lead to multicellular life. As the oceans continued to shift, some cells developed more stable methods of remaining together. Biofilms were among the earliest of these cooperative structures. A biofilm is a thin layer of cells adhering to a surface, perhaps a mineral grain, a rock, or an underwater surface of hardened volcanic glass. Within these films, cells secreted sticky substances that helped them anchor themselves and one another. These secretions provided protection against currents, predators, and other environmental stresses. Over time, cells in a biofilm found that they could persist in conditions that would otherwise have been too harsh for individual cells. Though each cell in a biofilm lived independently, the shared layer created a sense of community, a collective stability that benefited all who participated. Some early forms of cooperation arose not from adherence to surfaces but from the exchange of molecules. Certain prokaryotes released chemical signals into the water-simple compounds that diffused outward and could be detected by other cells of the same type. At first, these signals merely reflected the presence of cells in the area, but gradually, these chemical cues began to factor into cellular behavior. A cell might slow its movement when it sensed a high concentration of these molecules, indicating that many others were nearby. Or it might alter its metabolic rate, choosing to conserve energy or produce more nutrients in response to its neighbors. This primitive form of communication, though basic, allowed cells to adjust their activities collectively, improving their chances of survival. In these early interactions, we see the faint beginnings of a pattern that becomes essential to multicellular life. The idea that individual units can benefit by behaving in ways that support the group. The earliest cooperating cells were not yet forming true tissues or bodies, but they were practicing the underlying principles that would later allow such structures to exist. Cooperation also allowed early cells to occupy new environments. For example, some bacteria formed floating colonies that drifted on the ocean surface, held together by layers of secreted material. These colonies could remain in layers of water where sunlight was abundant, providing access to energy for photosynthesis. Cells deep in the colony benefited from the positioning of cells near the surface, and the entire community could remain suspended in the ideal region without sinking. Another form of cooperation emerged when some cells began sharing metabolic tasks. For instance, one cell might be particularly efficient at breaking down a nutrient into simpler components, while another cell specialized in converting those components into energy. When these cells lived close together, they could essentially divide the labor of metabolism, allowing both to thrive in environments where neither could survive alone. This division of labor was not yet organized or intentional, but it represented an evolutionary advantage. Simple cooperative communities of cells, each performing slightly different functions, formed the earliest prototypes for the specialized tissues found in multicellular organisms. Even the formation of stromatolites, layered structures built by microbial communities, reflects this early cooperation. These structures arise when microorganisms, especially photosynthetic bacteria, form layered mats that trap sediments and minerals over time. Stromatolites grew slowly but steadily, creating stable, protected environments for the cells within them. Their layered structure provided shielding from ultraviolet light, improved nutrient access, and offered a unique microenvironment where life could flourish in an otherwise challenging ocean. These ancient structures persist in the fossil record as some of the oldest evidence of life on Earth and stand as quiet monuments to the power of cooperation among simple cells. As the ages passed, cooperation deepened. Cells that communicated more effectively or adhered more firmly may have been more likely to survive environmental pressures. Slowly, evolution favored cells that could maintain stable associations for longer periods. These associations were not yet multicellular organisms in the way we understand them. The cells could still survive independently if separated, and there was no shared genetic control over their activities. Yet the seeds of multicellularity were present. Natural selection had begun shaping not just individuals but groups, turning simple clusters of cells into early collaborative communities. This journey toward cooperation did not happen in a rush. It was the product of millions upon millions of years, shaped by slow changes in ocean chemistry, climate, currents, and light. During this time, prokaryotes continued to diversify, experimenting with new ways of living together. Some remained solitary, finding success in independence. Others embraced the advantage of community, forming the earliest biological collectives. The ocean became a canvas where life painted its first patterns of connection, each small brushstroke contributing to the vast mural of evolution. Eventually, the patterns of cooperation would extend even further, influencing the rise of eukaryotic cells, the development of symbiotic relationships, and the eventual leap into true multicellularity. But at this early stage, the gift of cooperation was enough. It introduced stability into a world of constant change. It allowed simple organisms to explore new environments with confidence, and it provided the first gentle steps toward the intricate webs of interaction that sustain all living things today. As we move onward in our exploration, we carry forward the quiet truth that life does not grow more complex through sudden leaps. It unfolds through tiny, patient acts of connection, acts that began with the earliest prokaryotes drifting in ancient seas, discovering that it was sometimes easier to live not alone, but together. 5. The rise of eukaryotic cells. To understand the deep origins of multicellular life, we must pause at one of the most transformative thresholds in the history of biology: the emergence of the eukaryotic cell. This shift did not arrive quickly, nor did it appear in a sudden flash of complexity. Instead, it unfolded through countless small exchanges, through patient cooperation among simpler cells, and through the slow refining of structures that allowed life to hold more possibility within a single boundary. The rise of eukaryotes represents a gentle yet profound broadening of what life could be, providing the foundation for organisms with tissues, organs, and intricate systems. Before multicellularity could emerge, individual cells needed to become capable of greater internal organization, and this quiet transformation took place long before the first animals or plants came into existence. In the early oceans, prokaryotes thrived for vast spans of time, occupying environments rich with minerals and energy. They had simple structures, no nucleus, no membrane-bound organelles, and relatively modest genetic material floating freely within the cell. These forms were remarkably successful, and even today, bacteria and archaea are among the most abundant and resilient organisms on Earth. Yet somewhere within this ancient world, certain cells encountered opportunities for cooperation, so close and beneficial that they began to fuse their lives together in intimate, enduring ways. This process, known as endosymbiosis, was not a single event but a gradual weaving of existence between separate organisms. A larger cell may have engulfed a smaller one, not as prey but as a partner, and over time, these internal partners provided advantages too valuable to relinquish. One of the most significant results of this symbiotic union was the development of mitochondria, small organelles capable of producing large amounts of energy. The prevailing scientific understanding suggests that mitochondria originated from free-living bacteria that became integrated into early eukaryotic cells. Instead of being digested, these bacteria remained inside the host cell, offering their metabolic efficiency in exchange for protection and stable resources. Over long stretches of evolutionary time, the two became inseparable, each contributing to the survival of the other. Eventually, the engulfed bacteria transferred much of their genetic material to the host cell's DNA, losing their independence but gaining a secure existence within a larger system. This gentle merging of identities created a new kind of cell, one capable of supporting larger size and more complex functions than any prokaryote could manage. Another essential symbiotic event occurred in the ancestors of plants and algae. Certain eukaryotic cells formed partnerships with photosynthetic bacteria, cyanobacteria, leading to the creation of chloroplasts. These organelles enabled cells to capture sunlight and convert it into chemical energy, opening the door for life to spread across bright, shallow seas and eventually onto land. Chloroplasts, like mitochondria, retain traces of their bacterial origins. They carry their own DNA and reproduce within the cell in a manner reminiscent of ancient prokaryotes. These clues serve as gentle reminders of the ancient partnerships that transformed life on Earth and expanded its potential in ways that still echo through every leaf, every strand of algae, and every ecosystem reliant on photosynthesis. With mitochondria and chloroplasts in place, early eukaryotic cells gained tremendous advantages in energy production. This abundance of energy allowed them to grow larger, create more internal structures, and maintain more complex biochemical processes. The result was a cell with a nucleus, an enclosed compartment where DNA could be stored safely, and an array of organelles that performed specialized tasks. These included the endoplasmic reticulum, involved in protein and lipid synthesis, the Golgi apparatus, which packaged and delivered molecules, and various vesicles that transported materials around the cell. Together, these structures created an internal organization that mirrored, in miniature, the division of labor seen much later in multicellular organisms. The emergence of a nucleus was particularly important. In prokaryotes, DNA floats freely within the cytoplasm, exposed to fluctuations and disturbances. In early eukaryotes, DNA became enclosed within a double membrane, protecting it and allowing for greater control over how genes were expressed. This new layer of regulation made it easier for cells to adapt, evolve, and diversify. The nucleus provided a stable environment where genetic material could be duplicated accurately and transcribed efficiently. This refinement opened the way for more complex genomes, enabling cells to store more information and produce a wider variety of proteins. It is in these tiny, carefully safeguarded strands of DNA that the potential for multicellularity first took shape, even if the organisms of that time had no awareness of what their futures held. Eukaryotic cells also developed cytoskeletons, networks of protein fibers that provided structure and allowed cells to change shape, move materials internally, and even move themselves. This flexibility made it possible for cells to explore new environments and adopt new forms of behavior. For example, some early eukaryotes used their cytoskeletons to engulf food particles, a process called phagocytosis, which allowed them to access nutrients unavailable to simpler cells. This introduced new ecological roles, expanding the range of possible lifestyles and interactions. The simple ability to reshape themselves or extend parts of their cell bodies also marked a subtle but important step toward future multicellular coordination, where the shape and movement of cells would play critical roles in development. As eukaryotic cells diversified, some began forming colonies. These colonies were not yet true multicellular organisms, but they represented an evolutionary middle ground between solitary existence and integrated cooperation. In these groups, cells often performed similar tasks, but remained physically connected, sharing nutrients and responding to environmental cues collectively. Over time, certain colonies began displaying early forms of specialization. A few cells might become more effective at reproduction, while others took on supportive roles such as movement or structural stability. These changes, though small, the basics of tissue organization, cells taking on different responsibilities while maintaining unity, were already beginning to appear. Within these early colonies, communication became increasingly important. Eukaryotic cells developed more elaborate signaling pathways, using chemical messengers to coordinate activities both within and between cells. These signals allowed cells to respond to their neighbors, adjust their behavior when resources became scarce, or synchronize processes like reproduction. This growing ability to communicate added a new layer of complexity, making cooperative living more efficient and stable. Even though these early signals were simple compared to the intricate communication systems of modern multicellular organisms, they represented the gentle beginnings of cooperation at a more sophisticated level. Another crucial aspect of eukaryotic evolution was the development of sexual reproduction. Unlike the simple division of prokaryotic cells, sexual reproduction combined genetic material from two parent cells, introducing greater variation into populations. This increase in variation made it easier for natural selection to work effectively, allowing populations to adapt more quickly to changing conditions. Over long periods, this process accelerated evolutionary innovation, creating fertile ground for new forms of life to arise. Sexual reproduction also encouraged the formation of temporary multicellular structures in some early organisms, as cells sometimes touched or combined in ways that foreshadowed the more permanent unions seen later. All these developments, the presence of organelles, the protection of a nucleus, the structure provided by cytoskeletons, the beginnings of communication, and the introduction of sexual reproduction, worked together to create cells that were not just more complex, but more capable. They were equipped to handle larger genomes, more intricate chemical networks, and more coordinated interactions. Eukaryotes represented a profound shift, not only in structure but in potential. They carried the capacity to become something more than single cells. As time passed, this potential slowly unfolded. Certain lineages of eukaryotes remained solitary, thriving as independent cells. Others moved toward more cooperative lifestyles, forming the earliest multicellular organisms. These early steps were quiet, gentle, and experimental, carried out not with intention but with the slow guiding influence of natural selection. The rise of eukaryotic cells was not merely a biological event but a transformation in how life could be arranged, how functions could be shared, and how complexity could blossom from simplicity. As we prepare to move into the next part of our exploration, we carry with us the understanding that multicellularity did not arise from nothing. It grew from the delicate partnerships and subtle innovations within eukaryotic cells. Cells that learned to house internal communities and to cooperate with one another in deeper ways. From these humble beginnings came all the diverse forms of life we know, each one a testament to the quiet power of cells learning, slowly and patiently, to live not only as individuals, but together. 6. Why cells chose to stay together. In the long and gentle unfolding of life's early history, there came a time when individual cells began discovering that there were quiet advantages to remaining near one another. This shift did not happen with intention or planning, nor did it arise from any awareness within the cells themselves. Instead, it emerged gradually, shaped by currents, nutrients, and the subtle pressures of a changing planet. The earliest multicellular organisms did not appear suddenly. Their origins lie in the soft, Simple observation expressed not in thought but in survival that sometimes it was a little easier to live when one was not entirely alone. The slow transition from solitary life to collective living represents one of evolution's most profound transformations, yet it unfolded with a calmness befitting the ancient seas in which it began. When we imagine early cells drifting through warm oceans, we picture tiny individual worlds, each self-contained and complete, carrying out its own processes of metabolism, growth, and division. These early cells had everything they needed to survive independently, and for millions of years, many of them lived exactly that way. But the environment around them was not constant. Oceans changed in temperature and chemistry. Currents shifted nutrients from one region to another, and sunlight varied with depth and surface conditions. In such an unpredictable world, any slight advantage could help a cell survive a little longer, divide a little more often, and pass on its traits. And one of the most subtle yet powerful of these benefits was the simple act of staying close to others of the same kind. One of the earliest reasons for cells to remain together may have been purely physical. After a cell divided, the two daughter cells did not always separate cleanly. The membranes might remain lightly attached, or the surrounding water might be thick with dissolved minerals that slowed their dispersal. This gentle closeness offered no immediate change in the internal workings of the cells, but it did influence how they experienced their environment. A small cluster of cells could alter the flow of water around them, reducing turbulence and creating tiny pockets of stability. Within these pockets, nutrients might accumulate more easily, or harmful substances might diffuse away more gradually. In this way, proximity itself created a slightly more favorable microenvironment, an early hint of group advantage. As cells continued to divide without fully drifting apart, small chains or clusters formed. These early groupings had no specialized roles or coordinated behavior, yet they survived better than solitary individuals in certain conditions. For instance, in areas where sunlight reached only the upper layers of the ocean, clusters of photosynthetic organisms had a greater chance of maintaining position near the surface. A single cell, light and vulnerable, could easily sink or be swept downward by currents, but a cluster had more surface area, allowing it to remain buoyant for longer periods. This ability to stay within the sunlight allowed these cells to produce more energy, divide more often, and leave behind more descendants. Such simple physical benefits helped reinforce the value of staying connected after division. Another subtle reason cells began sticking together involved protection. The early Earth was filled with harsh conditions, shifts in salinity, fluctuations in temperature, ultraviolet radiation from a sun unfiltered by a modern ozone layer, and the presence of reactive chemical compounds. A lone cell exposed directly to these conditions might not survive, but cells within a cluster could shield one another. The outermost cells absorbed the brunt of environmental stress, while those deeper in the cluster remained insulated. This layering effect meant that clusters survived in environments where solitary cells could not. Even without intent, evolution rewarded cells that displayed a tendency to remain connected for longer periods. Chemical communication, which began as simple signaling between nearby cells. Many early cells released molecules into their surroundings, byproducts of metabolism or signals that alerted neighboring cells to changes in the environment. As these molecules diffused outward, cells nearby could sense them and adjust their behavior. Cells that remained close together could benefit from a shared understanding of local conditions. They might slow their metabolism if resources diminished, or they might increase certain processes when nutrients became abundant. These early forms of communication did not require consciousness, but they still created a subtle cohesion within groups. Over time, cells that responded well to communal signals were more likely to survive and reproduce within clusters. Another factor influencing the evolution of clustering was efficiency and resource use. Some cells were better at collecting certain nutrients or breaking down specific compounds, while others excelled at converting these compounds into usable energy. In groups, these differences created a tiny form of division of labor. One cell's byproduct became another cell's resource. This quiet exchange strengthened the stability of early clusters, making it less likely that individuals would drift away from one another. While not yet true specialization, this sharing of tasks represented a small but important step toward the more complex forms of division of labor that would later characterize multicellular organisms. As clusters grew larger, their internal dynamics became more intricate, even though each cell still behaved autonomously. The edges of these clusters absorbed nutrients and light, while the interiors remained sheltered. Cells within had a different experience of the world, calmer, more buffered, and more stable. Although the cells at the edge faced greater stress, they also had greater access to energy sources, enabling them to grow and divide. When they did, new cells often settled inward, contributing to a structure that was more stable and layered than anything seen in solitary life. With every generation, clusters that maintained their integrity for longer periods had a selective advantage. Eventually, some clusters became more permanent. Instead of drifting apart after a few cell divisions, they remained linked through subtle modifications to their cell walls or membranes. These modifications were simple at first, slight changes in the proteins or sugars that compose the outer layers of the cells. Over many generations, cells that produced these sticky or adhesive molecules persisted in clusters more reliably than those that separated. Natural selection quietly favored these traits, reinforcing the tendency toward cooperation. The earliest multicellular organisms may have arisen through such gradual reinforcement, guided not by any foresight, but by the steady rewards of remaining together. Some of the strongest early forms of cooperation developed through endosymbiosis, where one cell lived inside another. Although this is a more intimate form of cooperation, it reflects the same principle. Life began finding ways to benefit from closeness. Over time, these internal partnerships produced organelles like mitochondria and chloroplasts, structures that dramatically increased cellular efficiency. These innovations added further incentive for cells to explore new forms of association, ultimately creating a world where multicellularity was not only possible but increasingly favored. Multicellularity did not appear fully formed. It began with small steps, clusters that remained intact through several generations, slight variations in adhesion molecules, cells beginning to influence one another through chemical cues, gentle periods during which environmental stability rewarded cooperative living. Eventually, certain clusters of cells developed consistent patterns of growth, with some cells dividing inward and others outward, giving rise to simple body plans. Though these early forms were still far from the complexity of modern animals and plants, they demonstrated that cells could exist not just near one another, but as parts of a unified whole. As we reflect on these early developments, we begin to see how cooperation became a powerful force in evolution. Cells did not choose cooperation in any conscious sense, but their survival favored traits that supported togetherness. Over time, these traits deepened and diversified, guiding life gently toward the path of multicellularity. This shift set the stage for the vast diversity of creatures that would eventually populate Earth, from simple algae to towering trees, from soft-bodied organisms to the intricate forms of mammals and birds. In this calm and ancient story, we find the quiet truth that complexity grows from connection. Life becomes richer not through struggle alone, but through the slow, steady weaving of relationships that began with simple cells drifting in ancient seas. There was strength in staying close. 7. Communication. The first cellular conversations. In the long and patient unfolding of life's early history, there came a time when cells, drifting gently through ancient oceans, began to respond not only to the world around them, but also to one another. This shift was quiet, subtle, and entirely without intention. Yet it marked one of the most meaningful steps toward multicellularity. Communication among cells began not with spoken language or complex signals, but with the faintest chemical exchanges. Tiny whispers through the water that carried information from one living being to another. As these early conversations grew more refined, They set in motion the delicate threads of coordination that would eventually allow cells to live, work, and grow together as unified organisms. To imagine this early communication, we must first picture the world in which it began, a world of warm seas and scattered particles, where solitary cells drifted in enormous numbers. Each cell was small, yet busy with its internal rhythms, moving molecules, converting energy, maintaining its structure, and dividing when the moment was right. These cells lived simple lives, but they existed within shared environments, surrounded by countless others carrying out the same activities. It was natural, though unintended, that their chemical byproducts mixed into the surrounding water. At first, these molecules served no purpose beyond their immediate biochemical roles. They were simply released as cells metabolized sugars, responded to light, or rid themselves of waste. Over time, however, other cells began to detect these chemicals. A cell drifting near another might sense a change in the concentration of certain molecules. Perhaps the presence of another cell meant that nutrients were becoming scarce, or that conditions were changing. This early detection was not communication in the way we think of it today, but it formed the quiet beginning of a long evolutionary process. When a cell altered its behavior in response to chemical cues left by others, a new dynamic emerged, one where cells were no longer isolated in their experience of the environment. They began to influence one another, gently, through the natural currents of the sea. As cells evolved, their sensitivity to chemical cues increased. Some developed receptors, specialized proteins that allowed them to bind certain molecules and respond in predictable ways. When a particular molecule became abundant in the water, a cell might slow its metabolism to conserve energy or move to a different location where conditions were more favorable. These responses were simple, but effective. They gave cells the ability to adjust their behavior in relation to others, reinforcing early forms of cohesion. Gradually, the ability to sense and respond to chemical signals became a significant evolutionary advantage. In some early microbial communities, communication took on clearer patterns. Groups of cells released molecules that signaled population density, allowing them to regulate their collective behavior. When enough of these signals accumulated, cells shifted into new modes of activity, forming protective layers, adjusting their growth rate, or producing substances that help them adhere to surfaces. This phenomenon, known today as quorum sensing, reflects one of the earliest forms of true communication among living cells. Through this shared chemical awareness, cells began acting less like isolated individuals and more like members of a loosely organized community. Though still far from multicellularity, these interactions introduced a new level of coordination that would echo through all later stages of evolution. The development of internal cellular complexity also enhanced communication. In eukaryotic cells, the presence of internal membranes and organelles created opportunities for more refined signaling pathways. These pathways allowed cells to regulate their internal activities more precisely and to integrate multiple signals from their environment. Some signals encouraged cells to divide, while others led them to pause or to prepare for difficult conditions. Over time, these pathways allowed cells to interpret not just the presence of chemical molecules, but their concentration, timing, and source. As cells became more skilled at interpreting signals, their capacity for coordinated behavior increased. Communication between cells also played a role in forming colonies, where cells lived in consistent proximity. In these colonies, cells began responding to shared cues about nutrient availability, light intensity, and stress. When one part of a colony experienced stress, the cells in that region might release molecules that diffused outward, prompting neighboring cells to adjust their behavior. This diffusion created a soft wave of responses, allowing cells in different parts of the colony to act in ways that benefited the group as a whole. Over long periods, this kind of coordination shaped the earliest foundations of tissue-like structures, where cells align themselves according to both physical and chemical cues. An important aspect of early communication was the beginning of shared timing. In solitary cells, processes like division occur independently, but in colonies, the synchronization of these processes offered distinct advantages. For example, if many cells divided at once, they could reinforce the stability of the colony by producing new cells that filled in gaps or extended the structure. Synchronization also made it easier for colonies to maintain balance and respond to environmental changes. Although early synchronization did not require a formal leader or centralized control, the repeated sharing of chemical signals created rhythmic patterns in which many cells acted in harmony with their neighbors. Another gentle step toward communication involved self-recognition. Cells began producing molecules on their surfaces that allowed them to distinguish between their own kind and different types of cells. This ability helped colonies remain composed of compatible members and allowed cells to identify partners for cooperation. Over time, self-recognition became crucial to preventing unwanted fusion or competition within clusters, helping maintain the stability of early multicellular forms. It also laid the groundwork for more advanced systems of identity that would become essential in plants and animals. As communication grew more refined, the relationships between cells deepened. Some cells became more responsive to signals than others, allowing them to take on early leadership roles within groups. For instance, a cell might detect a shift in nutrient availability before others and send out signals prompting its neighbors to adjust their behavior. These subtle differences in sensitivity introduced the first glimmers of specialized roles. Although all cells remained capable of performing the same basic tasks, some cells became better at sensing, some at producing signals, and some at responding. This gentle diversification foreshadowed the division of labor that would later become central to multicellular organisms. The oceans of early Earth were vast, but communities of communicating cells created pockets of organized activity within them. Tiny landscapes, shaped not just by water and sunlight, but by biology's emerging capacity for shared information. These early conversations helped cells navigate uncertain conditions, locate nutrients, and avoid dangerous regions. They also allowed cells to grow in more coordinated ways, forming early cooperative structures whose persistence laid the groundwork for more advanced forms of life. The move toward multicellularity required not only that cells remain together, but that they communicate in ways that supported unity. Without communication, a cluster is simply a group of independent cells occupying the same space. With communication, a cluster gains the ability to act as a single, coherent whole, each part aware, in its own quiet way, of the others. This awareness was still primitive, built from the interplay of molecules drifting through water, but it was enough to support the next steps in evolution. As we reflect on these early cellular conversations, we see how gently cooperation began. No cell intended to communicate, yet communication emerged as a natural consequence of living together. It gave life a new dimension, allowing groups to behave in ways that solitary organisms could not. This soft, early exchange of signals was the first whisper of the complexity to come, a whisper that would carry life toward the deeper cooperative structures of tissues, organs, and eventually fully formed multicellular beings. 8. Division of labor begins. In the gentle, unhurried story of life's earliest transformations, there arrived a moment when the simple act of cells living together began to open doorways into new possibilities. At first, cells that remained in clusters behaved much like solitary individuals. They divided, absorbed nutrients, and responded to their surroundings in nearly identical ways. But over long stretches of time, the quiet influence of natural selection began shaping their interactions in subtle yet meaningful directions. The presence of nearby cells created gradients of light, nutrients, and chemicals that were not uniform across the cluster. As a result, some cells experienced the world differently from others, even when they belonged to the same tiny community. From these small differences arose one of evolution's most significant innovations: the early division of labor. To imagine the earliest stages of this process, picture a small cluster of simple cells drifting in warm, shallow water. Those on the outside of the cluster were bathed in light, in direct contact with the surrounding nutrients, and exposed to the fluctuating currents. Those deeper within experienced a calmer environment, more protected, but also more limited. The outer cells, with abundant access to resources, could divide more rapidly or produce molecules that benefited the colony. The inner cells, receiving fewer nutrients, might slow their division or become more efficient in conserving energy. Over countless generations, these incidental differences laid the foundation for specialization. This early form of division of labor was not deliberate. No cell chose its position or its role. Instead, roles emerged naturally from environmental variation. Yet, from the perspective of evolution, any arrangement that improved the survival of the colony tended to persist. Clusters that navigated their environments more effectively survived environmental shifts, enabling them to grow larger or produce more offspring clusters. These surviving groups passed along traits that supported a loose form of teamwork. Without conscious thought or intention, natural selection rewarded the faint beginning of coordinated function. As time passed, the chemical communication systems described earlier became more refined, enabling cells and clusters to sense not only their surroundings but also the activities of their neighbors. This improved the efficiency of resource use. For example, when nutrients became scarce, cells in one part of the cluster might release molecules signaling the need for conservation. Neighboring cells could respond by slowing metabolism or dividing less frequently, allowing the entire colony to endure longer periods of hardship. The ability to create shared responses gave these clusters a stability that solitary cells could never achieve. Eventually, subtle genetic differences arose within groups of cells. Some mutations affected how strongly cells adhered to their neighbors, how quickly they absorbed nutrients, or how efficiently they carried out certain tasks. When such mutations improved the colony's overall survival, they persisted through natural selection. Over great expanses of time, the combination of environmental gradients, communication cues, and genetic variation gently shaped the clusters into forms where roles began to diverge more distinctly. In some colonies, outer cells evolved to become more specialized in capturing energy. These cells absorbed sunlight and transformed it into the chemical fuels needed by the group. Meanwhile, protected inner cells, less exposed to light, took on different tasks, such as storing nutrients or dividing to maintain the structure of the colony. This early division of functions laid the groundwork for the later differentiation seen in multicellular algae, and eventually in the more intricate tissues of plants. Another important development occurred when some cells began adopting sacrificial roles. If the stability of a cluster depended on the adhesion between its cells, some cells might evolve to produce more adhesive substances, making them more strongly attached to their neighbors. These cells might divide less often, but contributed to the structural integrity of the group. Others nearby could benefit from the stability these adhesive cells offered, allowing them to reproduce more frequently. In time, both roles became essential to the survival of the collective. The emergence of structurally supportive cells foreshadowed one of the fundamental distinctions in multicellular organisms: cells that serve the body without directly producing offspring. Not all early clusters became more complex. Many remained simple aggregations that dissolved when conditions shifted. Yet in lineages where environmental pressures favored stability, early forms of specialization deepened slowly but steadily. Over time, entire colonies began behaving as cohesive units. When parts of a colony detached, they formed new clusters carrying the beginnings of role differentiation within them. Each generation reproduced not only its genetic material but also the spatial patterns and cooperative tendencies that allowed specialization to continue. One of the most compelling examples of early division of labor arises in modern organisms that occupy middle ground between single-celled life and full multicellularity. Some types of algae, for example, form spherical colonies with hundreds or thousands of cells arranged in layers. The cells on the outside of these colonies are often equipped with structures that allow them to move or steer, while the interior cells may focus on growth or reproduction. These organisms are living demonstrations of life's early steps toward specialization. They remind us that evolution often works through gentle transitions, not abrupt leaps. In other lineages, early specialization involved reproductive roles. Some cells began devoting themselves almost entirely to creating offspring, while others supported the colony's growth and movement. This separation between reproductive and non-reproductive cells mirrors one of the most fundamental divisions in multicellular organisms: the distinction between germ cells, which give rise to future generations, and somatic cells, which support the structure and function of the body. In these ancient clusters, the groundwork for this distinction began quietly and organically, shaped by simple environmental pressures and long spans of evolutionary time. As communication pathways became increasingly elaborate, cells gained the ability to produce signals that directly influenced the fate and behavior of neighboring cells. These signals gradually formed the earliest developmental patterns. A cell located near a source of certain signals might adopt a role of structural reinforcement, while another located farther away might be more likely to divide or to generate movement. Each cell's role became a gentle reflection of its position within the colony and the chemical messages it encountered. These early forms of regulation foreshadowed the processes that guide modern development, where chemical gradients direct cells to become muscle, bone, nerve, or skin. Through this slow, steady process, life began organizing itself into structures where no single cell carried the entire burden of survival. Instead, different cells contributed in different ways, creating an early harmony of roles. The division of labor made it possible for clusters to grow larger, to occupy more stable environments, and to respond more effectively to challenges. It also set the stage for the evolution of true multicellularity, where cells no longer simply lived together but became interdependent parts of a larger whole. In the soft rhythms of these early oceans, the first faint outlines of tissues and organs began to appear, not as complete systems but as tendencies, cells behaving differently based on their surroundings and interactions. This gentle differentiation was one of life's quiet triumphs, an innovation that would eventually allow organisms to develop astonishing complexity. Yet at this stage, all of it was still simple, peaceful, and unhurried, driven not by ambition but by the calm movement of water, the quiet exchange of molecules, and the slow shaping hand of natural selection. As we prepare to move forward, we carry with us the understanding that division of labor arose not through sudden invention, but through millions of subtle moments in which cells adapted to their surroundings and to one another. In this way, life's earliest communities began to resemble more than simple clusters. They became coordinated collectives, capable of paving the way for the elegant intricacies of true multicellular existence. 9. Early multicellular landscapes of the sea. In the quiet expanses of Earth's ancient oceans, long before forests rose on land or animals swam with purposeful motion, the earliest multicellular forms began to appear. Their emergence did not happen with sudden flourish. It unfolded gradually. These early organisms were gentle presences, soft-bodied, delicate, and often so simple that they resembled living shadows more than creatures. Yet they marked the beginning of a new era in the history of life, an era when individual cells were no longer the highest level of biological organization. The sea began to hold not just solitary beings or loose clusters, but unified bodies composed of many cells working together in quiet harmony. To imagine these early landscapes, let us drift back into the calm, ancient seas where life's complexity was just beginning to deepen. These oceans were warm and rich with minerals. Their shallow regions glowed softly beneath the filtered light of a young sun, creating an ideal stage for life to experiment with new forms. After millions of years of gradual change, some clusters of cells had learned to remain together in stable configurations. Their connections, once temporary, had become more lasting, supported by proteins and sugars on the surfaces of their membranes. This stability allowed these clusters to persist long enough for natural selection to guide their development, gently shaping their growth patterns and internal organization. The earliest multicellular organisms were probably modest in form, simple filaments, sheets, and spheres. Imagine thin strands of cells stretching across the water, each cell connected to the next like beads on a soft, living thread. These strands might have anchored themselves lightly to underwater surfaces, such as rocks or patches of sediment. As they grew, the filaments expanded into graceful, wispy shapes that swayed with the currents. The ocean's movement did not harm them. Instead, it helped them disperse nutrients, maintain their positions, and release new cells that could drift away and settle elsewhere. Other early multicellular organisms formed flat, leaf-like sheets. These sheets were composed of layers of cells stacked gently atop one another, creating larger and more stable structures than single-celled forms could achieve. Because the cells on the surface had greater access to light, they often became more specialized in capturing energy, while the cells beneath served structural or reproductive roles. These sheets rested quietly on the seafloor or floated slowly near the surface, creating carpets of living tissue that responded to the sun's warm guidance. They were simple, yet elegant, offering one of the first hints at how multicellular forms might diversify into broader and more complex shapes. Still, other organisms formed spherical structures, with cells arranged in shells or clusters around inner cavities. These spheres had many advantages. They protected the interior cells, allowed water to circulate through their open spaces, and created small environments with greater stability than the surrounding ocean. Some spheres developed the ability to rotate or move slowly through the water by coordinating the movement of small hair-like structures on their outer surfaces. Though their movements were subtle, they marked important beginnings. The ability to shift position, even slightly, gave these organisms greater control over where they lived, allowing them to find pockets of light, warmth, or nutrients more easily. The early multicellular seas were calm worlds of gentle forms, but they were not uniform. In some regions, clusters of cells lived in colonies where a degree of specialization had begun to emerge. In others, organisms were more loosely organized, with cells that could still survive independently if separated. Life was experimenting across a vast range of possibilities, testing how different structures and connections might influence survival. The transition from single-celled to multicellular existence was not a single path, but a wide array of branching explorations. Some branches led to simple dead ends, organisms that were stable but not adaptable. Others continued to evolve, creating forms that could withstand changes in current, temperature, and nutrient availability. In these seas, stromatolite structures formed by layered bacterial communities still played a significant role. Though stromatolites were not multicellular organisms themselves, they created complex habitats that allowed early multicellular forms to flourish. Their surfaces provided anchoring points for filaments and sheets. Their layers created gradients of light and chemistry that supported different types of life. In this way, the microbial landscapes of the early oceans nurtured the emergence of more complex beings, offering shelter and gentle protection within their layered forms. As multicellular organisms grew more stable, their bodies began to exhibit the faintest hints of organization that would one day lead to true tissues. Cells in different regions of the organism experienced different conditions. Some received more light, some more nutrients, some more contact with neighboring cells. These subtle differences gradually shaped variations in cell behavior. Over generations, some cells became more specialized in absorbing energy, while others became more efficient at dividing or defending the organism's structure. Even though these early forms lacked organs or dedicated systems, the early patterns of specialization were gently taking root. Life was beginning to recognize through natural selection alone, that doing different tasks in different regions of the body could lead to greater overall stability. The early multicellular seas were places of quiet change. The increased size of multicellular organisms offered protection from environmental fluctuations, making them more resilient to currents, temperature shifts, and variations in nutrient supply. Larger bodies also created internal environments that allowed cells to exist in conditions more stable than those found outside. This enabled cells to perform their functions without being constantly exposed to the full volatility of the ocean. Over time, this internal stability paved the way for more complex interactions among cells and for more refined patterns of development. Another gentle advantage of multicellularity came from reproduction. Some early multicellular organisms reproduced by releasing clusters of cells rather than single ones. These clusters already had the beginnings of coordinated function, making it easier for them to establish themselves in new habitats. This provided a way for life to spread across broader regions of the ocean, colonizing new areas and expanding its presence. The sea, once populated only by solitary cells, slowly filled with softly drifting bodies of many shapes, each representing a new experiment in collective life. As multicellular forms became more widespread, they also modified their environments. They created new surfaces for microorganisms to live on, influenced the chemistry of the surrounding water, and altered the flow of nutrients through the ocean. These subtle environmental changes, though gentle, contributed to feedback loops that further encouraged the evolution of complexity. In this way, multicellular organisms did not simply arise within the ocean, they helped reshape the ocean itself. In the stillness of these early seas, life was beginning to explore the idea of being many cells rather than one. Simple though they were, these early multicellular organisms laid the foundation for everything that would come next. They showed that life could grow larger, more organized, and more interactive without sacrificing the calm equilibrium that had always been essential to its survival. They prepared the way for the next, more intricate steps, structures, more complex, interactions deeper, and the gradual appearance of true tissues and small, coordinated bodies. As we continue moving forward on this soft evolutionary path, we will enter the stages where multicellular organisms begin to explore more sophisticated ways of living, ways that include capturing energy more efficiently, interacting with their environment in coordinated ways, and forming the first primitive body plans. But for now, we pause with these early pioneers, drifting in their gentle landscapes, unaware that their quiet existence was shaping the future of life on Earth. 10. The influence of light and energy. In the gentle progression of life's early history, sunlight played one of the most essential and calming roles. Long before animals learned to swim or plants lifted leaves toward the sky, light moved slowly across the surface of the ancient seas, warming the shallows and guiding the rhythms of primitive organisms. In these quiet waters, the first multicellular beings began to sense and respond to energy in ways that helped shape their growth, their structure, and their long-term evolutionary path. Light was not a force of urgency or disruption. It was a soft presence, steady, predictable, and nurturing, providing a source of energy that made the early oceans a cradle for life's expanding complexity. To understand how light influenced early multicellular life, we must first imagine the nature of Earth's ancient sunlight. The atmosphere was not yet like our own. Oxygen was still limited, and the protective ozone layer had not fully formed, allowing a mixture of gentle warmth and more intense radiation to reach the ground and the sea. Yet in the upper layers of the ocean, water acted as a natural filter, softening the harsher rays while allowing the useful wavelengths of light to pass through. In this band of illuminated water, neither too shallow nor too deep, life found an ideal environment for photosynthesis. It was in these softly lit zones that early multicellular organisms began to flourish, developing new strategies for capturing and using energy. These early organisms were simple, often composed of only a few layers of cells, but their arrangement allowed them to gather light more effectively than solitary cells could. For example, organisms forming thin sheets or filaments could expose a larger surface area to sunlight, increasing the amount of energy they absorbed through photosynthetic pigments. This added energy allowed them to grow larger, maintain stable internal environments, and produce offspring more steadily. In a world where resources were unevenly distributed, the ability to capture sunlight consistently became one of the most gentle but powerful advantages that multicellularity offered. The relationship between light and multicellularity deepened as organisms began to differentiate. Cells on the surface of a colony or organism had better access to sunlight and thus became more active in energy production. Meanwhile, cells beneath the surface, sheltered from direct light, often shifted toward roles involving growth, repair, or reproduction. This natural gradient light on the outside, darkness within, encouraged the early forms of specialization that are essential to all modern multicellular life. Here, in the simple contrast between light and shade, nature began shaping the earliest tissues without conscious design. Light also influenced the physical structure of early multicellular organisms. Those living near the ocean surface often developed flattened or branching forms that spread their cells outward, maximizing exposure to sunlight. Branching shapes allowed them to anchor lightly while extending delicate filaments upward where light was stronger. Flat forms, such as thin sheets, could float or remain suspended in gently moving water where they received sunlight evenly. These shapes were not yet complex body plans, but they represented evolutionary solutions that improved the efficiency of light capture. Energy availability, particularly from sunlight, influenced where early multicellular organisms could survive. The photic zone, the upper layer of the ocean where light penetrated, became a region rich with life, while deeper waters remained the realm of simpler organisms relying on chemical energy. Over time, the abundance of multicellular life in illuminated waters created new ecological interactions. Organisms competed softly for space, for light-rich regions, and for nutrients carried upward by currents. Yet, because these interactions unfolded slowly over millions of years, they formed a calm evolutionary framework in which the diversity of multicellular organisms could expand. Sunlight also shaped the chemical composition of Earth's atmosphere and oceans. Photosynthetic organisms, both unicellular and multicellular, released oxygen as a byproduct of absorbing light. As oxygen accumulated, it created opportunities for new metabolic pathways. Organisms that evolved to use oxygen for energy gained access to far more efficient systems of metabolism. This made it possible to support larger bodies and more complex internal organization. Although the oxygenation of Earth was a slow and sometimes unstable process, multicellular life benefited deeply from its gradual rise. The increased availability of oxygen allowed tissues to form, grow, and persist in ways previously impossible. In addition to photosynthesis, the distribution of light shaped the daily rhythms of early multicellular life. Even simple organisms can respond to light and dark cycles. As Earth rotated, the soft pattern of day and night created predictable changes that early multicellular organisms learned to navigate. Many grew more rapidly during periods of abundant light and conserved energy when light faded. These gentle rhythms provided structure to their behavior, reinforcing the stability of their growth. Over time, these patterns became deeply embedded in the functioning of biological systems, influencing cycles of reproduction, movement, and metabolism. Light also contributed to the diversification of early multicellular organisms. Variations in light availability, whether due to depth, clarity, or geography, created different ecological niches. In clearer waters, organisms could spread out vertically, forming floating mats or suspended colonies. In murkier waters, organisms might remain closer to the surface or develop pigments to enhance light absorption. Some evolved deeper colors to trap light more efficiently, while others developed transparent tissues that allowed light to penetrate more deeply into their bodies. These soft changes in pigment and shape reflected the quiet but steady influence of sunlight across evolutionary time. Each adjustment allowed organisms to thrive in environments with slightly different light conditions, gradually shaping the diversity we glimpse in their ancient descendants. The relationship between multicellular life and light created a feedback loop that encouraged further complexity. As organisms grew larger and began forming more stable structures, they altered water flow and nutrient distribution. This, in turn, influenced where other organisms could receive light and energy. Some species began to rely more heavily on nutrients brought from deeper waters, while others adapted to the surfaces of rocks, stromatolites, or sheltered areas where light was filtered. Each of these shifts gently nudged evolutionary pathways in new directions, promoting both stability and change in the early multicellular world. As we reflect on the influence of light and energy on early multicellular life, we see that this relationship was not dramatic or sudden. It was a long, calm, shaping process like the slow warming of the ocean under the sun's gaze. Light did not force life into complexity. It invited life to explore new forms, offering energy in steady, gentle waves that supported growth, specialization. Through countless cycles of day and night, life learned to orient itself toward the sun, to spread outward, to divide roles among its cells, and to inhabit new environments made possible by light's quiet guidance. The early multicellular landscapes of the sea were peaceful scenes, filled with drifting forms and slowly growing colonies, all connected by the soft thread of sunlight. As we move onward, we will see how other environmental factors, nutrients, temperature, and ocean currents continued to shape life's unfolding, encouraging organisms to refine their cooperation and develop new strategies for survival. But for now, we rest in the understanding that light, with its calm constancy, was one of life's first great teachers, guiding the earliest multicellular beings toward the promise of greater complexity. 11. The role of nutrients and ocean mixing. In the quiet expanses of Earth's ancient oceans, life unfolded in rhythms shaped not only by light but also by the gentle movement of nutrients through the water. These nutrients elements such as nitrogen, phosphorus, iron, and carbon were the subtle threads that nourished early cells, and eventually, the first multicellular organisms. They drifted through the sea in slow cycles, carried by currents, upwellings, and the soft exchange between water layers. Long before complex ecosystems emerged, the availability of these nutrients determined where life could grow, how organisms adapted, and which multicellular forms found the stability needed to persist. The story of early multicellular life is inseparable from the calm flow of nutrients, for without these essential ingredients, the earliest experiments in complexity could not have unfolded. In the very beginning, nutrients entered the ocean from deep within the earth. Volcanic activity released minerals and gases that dissolved into seawater, enriching it with the raw materials needed for metabolic processes. Hydrothermal vents along the seafloor emitted warm, mineral-laden water, creating chemical gradients that nourished clusters of early microorganisms. These vents were like quiet oases, offering warmth and chemical energy in the darkness of the deep sea. Although these environments favored simpler organisms, they also contributed to the overall nutrient balance that influenced life in shallower waters where multicellularity would later take hold. The distribution of nutrients in the early ocean was far from uniform. Deep waters, protected from sunlight, were rich in nutrients carried down by sinking organic matter. As a result, life thrived most fully where these conditions overlapped places where nutrient-rich water rose toward the light. This rising, known as upwelling, was a gentle but powerful force. It brought essential nutrients from the deep ocean back into the illuminated zone where photosynthetic organisms lived. These upwellings were not sudden or violent. They were slow, rhythmic circulations shaped by currents, wind patterns, and variations in water temperature. Yet their influence on early multicellular life was profound. In regions where upwelling occurred consistently, early multicellular organisms found steady nourishment. Sheets of algae, delicate filaments, and spherical colonies floated or rested in these nutrient-rich zones, absorbing minerals that supported growth and reproduction. With access to abundant resources, these organisms could experiment with slightly larger sizes, more stable structures, and early forms of specialization. Nutrient availability allowed surface cells to focus on capturing sunlight while inner cells took on supportive roles. In this soft interplay between sunlight and nutrition, the foundations of early tissues were laid. Nutrient cycles also influenced the shape and behavior of early multicellular forms. Organisms in nutrient-poor waters often grew more slowly, remaining small and simple. Those in nutrient-rich regions could afford to invest energy in branching structures or expanded surfaces. This created a gentle gradient in multicellular complexity across the ocean. Shallow lagoons might host simple filaments, while coastal upwelling zones supported broader, more elaborate sheets of cells. In each region, life adapted quietly to the patterns of availability, forming early ecosystems that reflected the calm interplay between chemistry and environment. Another important source of nutrients came from the decomposition of earlier life forms. As cells died, their organic material sank or floated, breaking apart and releasing nitrogen, phosphorus, and other vital elements back into the water. This recycling created a slow, circular rhythm that sustained life. Even early multicellular organisms benefited from this natural cycle. As dead cells settled onto the seafloor or drifted into the deeper waters, bacteria broke them down, releasing nutrients that would eventually return to the surface through mixing. These cycles taught early life a gentle lesson, that even decay contributed to the persistence of the community. Seasonal variations also played a role in nutrient distribution. In some regions, changes in temperature between seasons caused layers of water to mix. Warm surface waters cooled and sank, while deeper waters rose. This turnover refreshed the photic zone with minerals, creating periods of enhanced growth. Early multicellular organisms responded to these cycles with subtle adjustments. During nutrient-rich periods, they expanded more rapidly, producing new branches, sheets, or spheres. During nutrient-poor seasons, they conserved energy, slowing growth and relying on internal reserves. These quiet adaptations allowed them to endure the natural rhythms of their environment, becoming part of the greater oceanic tapestry. Just as light created gradients that encouraged differentiation within organisms, nutrients created gradients across the ocean that encouraged diversity among them. Different species adapted to different nutrient conditions, creating gentle ecological niches. Some organisms became specialists in low-nutrient environments, evolving efficient methods of absorption or storage. Others flourished where nutrients were abundant, evolving larger bodies or more elaborate arrangements of cells. These early patterns of distribution formed the first soft outlines of ecological communities, clusters of species that shared habitats shaped by the same quiet flows of nutrients. As multicellular life continued to develop, interactions between organisms began influencing nutrient cycles more directly. Some early algae oxygenated the surface waters through photosynthesis. Others contributed organic matter that sank to the ocean floor, feeding deep communities that would later return nutrients upward through decomposition. Even simple multicellular organisms played their part. Their growth altered water flow around them, changed how sediments settled, and created small microenvironments where nutrients accumulated or dissipated. Each organism, by its mere presence, became part of a long and gentle conversation with the environment. Nutrient availability also encouraged some organisms to anchor themselves more firmly. While many early multicellular organisms drifted freely, others found stability by attaching to rocks, sediments, or the surfaces of stromatolites. At these attachment points, water often flowed steadily, bringing a constant supply of nutrients. In these stable habitats, Multicellular organisms developed broader surface areas and thicker cell layers. Anchored life gained the ability to occupy specific locations and form communities that interacted softly with the changing conditions around them. Changes in nutrient availability over long periods also played a role in natural selection. When nutrients were limited, organisms that used them more efficiently persisted. When nutrients were abundant, diversity blossomed, and new forms emerged. This balance between scarcity and abundance created cycles of innovation, periods in which new shapes and strategies developed, followed by times of consolidation and refinement. Although these changes occurred over millions of years, they were all tied to the gentle flow of nutrients through the ocean. The influence of nutrients extended into the earliest stages of developmental complexity. When clusters of cells began forming internal cavities, those cavities allowed water to flow through the organism's body. This flow brought nutrients to interior cells and carried away waste. Such early openings represented the first steps toward specialized body structures, such as simple digestive regions or internal channels. These innovations arose from the quiet relationship between cells and their need to access nutrients even as the organism grew larger. As we reflect on the early multicellular seas, we see a world shaped not by rapid events, but by slow, nourishing cycles. The mixing of deep and shallow waters, the rise and fall of seasonal temperatures, and the gentle decomposition of organic matter created a calm but dynamic environment where multicellular life could take its first steady steps. Nutrients moved through the ocean with patience. In this way, the sea itself acted as a soft guide, leading life toward greater complexity through the simple act of nourishment. Now, with this understanding of how nutrients shaped early multicellular evolution, we can continue gently into the next part, where temperature and other quiet force begins to guide life toward new adaptations and new forms. 12. Temperature as an evolutionary guide. In the soft and ancient waters where early multicellular life was unfolding, temperature acted as a quiet, persistent guide. It shaped the rhythms of the ocean, the movement of nutrients, and the behavior of cells long before any organism had the ability to sense or control its surroundings. Unlike abrupt forces such as storms or volcanic events, temperature changed slowly, creating gentle gradients across the sea that encouraged life to explore different forms and functions. In these warm and cool currents, early multicellularity found both its challenges and its opportunities. To imagine how temperature shaped the early oceans, it helps to picture Earth not as the familiar planet we know today, but as a younger and more dynamic world. The sun was slightly dimmer, the atmosphere still evolving, and the distribution of land and water very different. The oceans absorbed and released heat in broad, slow cycles that set the conditions for early metabolism. Because temperature influences the activity of enzymes, the tiny molecules that drive life's chemical reactions, even slight variations could change the pace of cellular processes. A modest increase in warmth could make reactions more efficient, while cooler waters might slow them down, encouraging cells to conserve energy or adopt different strategies for survival. These subtle differences influenced where multicellular life could take hold. In warmer, shallow waters, early organisms often found energy more abundant. Here, photosynthetic cells on the outer surfaces of simple multicellular bodies could capture sunlight more efficiently, supporting the growth of entire colonies. These warm zones became areas where diversity increased. Filamentous organisms extended themselves toward the light, forming mats that rested quietly on the seafloor. Spherical colonies drifted with slow currents, their outer cells absorbing energy while inner cells engaged in division or storage. The warmth of the environment gently encouraged faster metabolism, allowing these early forms to experiment with size, shape, and arrangement. In contrast, cooler waters offered different advantages. Although metabolic reactions slowed, cooler temperatures often preserved nutrients and oxygen more effectively. Some early multicellular organisms found stability in these regions, relying on slower, steadier growth rather than rapid expansion. Cooler environments also sheltered organisms from the harsher ultraviolet radiation that penetrated the shallowest waters in the absence of a fully developed ozone layer. This gave cells time to refine internal structures and communication systems without the constant pressure of rapid turnover. In these gentle cold zones, early lineages developed traits that supported resilience and long-term survival. Temperature gradients also influenced the behavior of cells within the same organism. As multicellular forms grew thicker or more complex, not all cells experienced the same temperature. Outer cells warmed more quickly under the sun, while inner layers remained cooler. These contrasts gently shaped early differentiation. Warmer cells might divide more often or produce energy more efficiently, while cooler cells became more specialized in conserving resources or maintaining the structural integrity of the organism, even without conscious control. These temperature-driven differences nudged cells toward distinct roles, an early whisper of specialization that would later define tissues. Ocean currents, influenced by temperature differences, played a crucial role as well. Warm water is lighter and tends to rise, while cooler water sinks. This created slow, steady circulations within the sea, carrying nutrients upward and distributing oxygen throughout the water. Multicellular organisms that positioned themselves in these circulating zones gained regular access to fresh supplies of dissolved minerals. Organisms attached to rocks or sediments in these areas benefited from the gentle flow, which brought nourishment to their surfaces. As a result, their growth patterns reflected the natural movement of the water. Some grew into branching shapes that maximized exposure, while others spread into broad, leaf-like forms that captured resources more efficiently. Seasonal variations in temperature added another layer to this evolutionary guide. As the planet tilted on its axis, some regions experienced cycles of warming and cooling. During warmer seasons, multicellular organisms grew more quickly, taking advantage of increased metabolic activity and nutrient availability. During cooler seasons, they slowed their processes, conserving energy and allowing their colonies or bodies to rest. This rhythm was not stressful, it was simply a gentle pattern that early life learned to follow. Over time, these repeated cycles encouraged the development of internal mechanisms capable of sustaining life through both abundance and restraint. Another subtle influence of temperature involved the viscosity of seawater. Warmer water is less dense and less viscous, allowing organisms to drift more freely. In such conditions, colonies that depended on floating or gentle suspension in the water column thrived. Their bodies could spread outward or extend upward without fighting the thick resistance of cooler water. In contrast, colder seas, with their higher viscosity, encouraged organisms to anchor themselves to surfaces. Attached forms, thin sheets, spreading filaments, or softly branching structures persisted in these environments because mobility was less advantageous. These slow adaptations gradually shaped different evolutionary pathways, guided by the temperature's influence on the texture of the sea. Temperature also played a role in the earliest interactions between multicellular organisms and microbes. Some bacteria thrived in warm, nutrient-rich waters, forming layers that early multicellular organisms learned to grow upon or near. Others were more abundant in cooler regions, where they contributed to nutrient recycling. These microbial landscapes created gentle gradients of opportunity. Multicellular life followed these gradients, anchoring itself where temperature conditions created pockets of ecological stability. Through this slow process, temperature helped define the earliest communities, simple but interconnected groups of organisms that shared space, resources, and chemical exchanges. Over long periods, temperature patterns across the planet shifted. Geological changes altered ocean currents. Volcanic activity warmed or cooled regions, and the gradual accumulation of oxygen in the atmosphere changed how heat was distributed. Each of these changes influenced life, but not in sudden ways. The early oceans responded slowly, maintaining their calm patterns of mixing and circulation. Multicellular organisms adapted over generations, refining their structures and internal processes in harmony with these changes. Life's earliest branching experiments, whether towards sheets, spheres, or filaments, were guided by the temperature patterns that shaped their environments. One of the most profound influences of temperature was its effect on developmental timing. In warmer waters, the steps leading to cell differentiation unfolded more quickly, allowing multicellular organisms to form organized structures in shorter periods. In cooler waters, these processes occurred more slowly, giving cells more time to interact, communicate, and stabilize. Faster development allowed organisms to respond quickly to favorable conditions, while slower development allowed for the refinement of early patterns that might otherwise have been lost through rapid turnover. In this gentle variability, temperature encouraged a diversity of strategies that enriched the early multicellular landscape. As we reflect on the role of temperature in early evolution, we see a world shaped not by abrupt transitions, but by gradual thermal rhythms. Warm zones nurtured rapid growth and experimentation, while cooler zones encouraged stability and precision. Together, these environments created the first mosaic of multicellular life, a tapestry woven from the soft gradients of sunlight, nutrients, and temperature. Each thread contributed to the growing complexity of organisms that were still simple by modern standards, but profoundly innovative compared to their ancestors. In moving forward, we will continue to explore how life learned to refine its forms. The next part will bring us deeper into the structures that early multicellular organisms began to build, the patterns and shapes that laid the foundation for true body plans. But for now, we rest with the understanding that temperature, gentle and omnipresent, guided life's early steps as softly as a warm current moving through an ancient sea. 13. Life learns to build patterns. In the calm progression of early evolution, once cells had learned to stay together, to communicate gently, and to share the work of survival, a new possibility began to emerge. Life started discovering the quiet art of arrangement. Grouped cells, no longer existing as loose, shapeless clusters, gradually began forming patterns simple at first, but filled with the potential to transform the living world. These patterns were not the result of intention or design. They arose naturally from the interactions of cells with their environment, from subtle gradients of light and nutrients, and from the early chemical signals that pass softly from one cell to the next. Through these influences, life's earliest multicellular forms began to take shape in ways that were increasingly organized, giving rise to structures that hinted gently at the future body plans of more complex organisms. To understand how these patterns formed, imagine a small cluster of cells resting in the stillness of an ancient shallow sea. The cells along the outer surface experience the world differently from those inside. Light touches them first, nutrients move past them, and currents influence their positions and movements. These outer cells, sensing the environment more directly, may grow or divide at different rates compared to their sheltered neighbors. Over time, these subtle differences create a slight asymmetry, perhaps a gentle elongation in one direction or a widening in another. Such changes are quiet and slow, but they accumulate. What was once a simple cluster becomes something more structured, a filament, a sheet, or a hollow sphere. These simple shapes formed the earliest patterns of multicellular life. Filamentous structures stretched outward, cell by cell, creating slender chains that anchored themselves lightly to surfaces or drifted through sunlight-filled waters. Sheets of cells extended across the seafloor like living carpets, maximizing exposure to the light needed for photosynthesis. Spheres created stable internal environments that sheltered inner cells while allowing the outer ones to interact more directly with their surroundings. Each shape represented a different solution to the same gentle challenge: how to organize many cells in a way that allowed them to thrive as one. Patterns emerged not only from environmental cues but also from the chemistry of life itself. Certain molecules on the surfaces of cells promoted adhesion, causing them to stick together in consistent ways. Some cells developed stronger adhesion on one side than the other, encouraging the formation of layers or curves. As cells divided, their daughters often stayed in predictable positions relative to their parents, creating repeating arrangements that persisted over time. These early rules of placement were simple, but as the number of cells increased, the arrangements they created became more elaborate. Even without conscious control, the patterns of cell division, movement, and adhesion began shaping early organisms with increasing regularity. Inside these simple bodies, communication played a growing role. As cells released chemical signals, those signals did not spread evenly. Instead, they formed gradients stronger in one region, weaker in another. Cells reacted to these gradients by adjusting their growth and behavior. If a signal encouraged division, cells in areas where the signal was strongest might divide more quickly, expanding that region. If a signal promoted stability or adhesion, cells exposed to it might form stronger bonds. These subtle variations guided the formation of patterns across the developing organism, creating regions with distinct identities even at very early stages of multicellularity. Interactions with the environment also influence pattern formation. Currents might push one side of an organism more than another, encouraging the development of structures that resisted or took advantage of that movement. In some regions, sunlight might hit one side more intensely, causing photosynthetic cells in that area to grow more actively. Nutrients might flow more freely across certain parts of the organism, leading to local areas of enhanced growth. All these influences contributed to the emergence of shapes that were not random but were instead gentle reflections of the conditions in which life existed. In filamentous organisms, patterns developed along a single axis. Cells divided in one direction more often than in others, producing long, thread-like bodies. These filaments sometimes branched, creating networks that increased their surface area. Branching was often triggered by a small imbalance, perhaps a nutrient-rich region on one side or a slight chemical difference between neighboring cells. Once branching began, it often continued at a measured pace, creating shapes reminiscent of delicate underwater plants. In sheet-like organisms, cells divided across two dimensions, creating broad, flat structures. Over time, these sheets could fold or curve, responding to currents or shifting sediments beneath them. Some sheets developed thicker central regions, where cells divided more frequently, giving the sheet a gentle dome-like shape. Others remained thin and flexible, adapting gracefully to the movements of the water around them. Spherical organisms develop patterns based on interior and exterior differences. Cells on the outside absorbed light and nutrients, while inner cells often became more reproductive or supportive. This separation created a natural division of roles that further encouraged patterned growth. Some spheres developed small openings that allowed water and nutrients to flow inside, creating early versions of internal cavities that would one day become common in more complex animals. Pattern formation did not occur only on the scale of entire bodies. Even within small clusters, cells began to show preferences for where they settled. Some adhered more strongly at the bottom, forming anchoring regions. Others gathered along the outer surface, forming protective layers. Interior cells arranged themselves where space allowed, often forming loose networks or gently packed groups. Although these internal patterns were simple, they represented the earliest hint that cells could organize themselves into differentiated structures that would eventually give rise to tissues. Over time, as multicellular organisms reproduced, they passed these patterns along to new generations. The rules of cell placement, division, and adhesion became encoded in their genetic material, allowing offspring to develop similar shapes. This stability allowed natural selection to influence not only individual cells, but the entire architecture of early organisms. Those that formed patterns well suited to their environments persisted and diversified. Those with less effective arrangements faded quietly from existence. The development of patterns also encouraged the emergence of symmetry, one of nature's most calming structural principles. Some organisms developed radial symmetry, forming circular or spiral shapes that functioned equally well in all directions. Others began forming early hints of bilateral patterns, favoring one direction of growth over another. These symmetries were not yet as defined as they would become in later animals, but they represented the first steps toward organized body plans. Through these soft adaptations, life began to explore forms that were both stable and aesthetically balanced. As we reflect on these early patterns, we see life taking its first quiet steps toward structured existence. The shapes that emerged in ancient seas were not complex, but they held within them all the potential for the sophisticated organisms that would come later. In simple filaments, sheets, and spheres, life learned the gentle art of arrangement, an art that would eventually lead to tissues, organs, and entire bodies shaped by the same forces that guided these earliest steps. With this calm foundation of pattern building, we are ready to move forward to the next part, where the first true tissues begin to form, deepening the harmony of multicellular life. 14. The first true tissues. In the gentle unfolding of early multicellular life, there came a moment when cells, once loosely connected and only softly specialized, began to work together in ways that formed the earliest tissues. These tissues were not yet like the complex muscles, nerves, or organs we see in modern animals. Instead, they were simple, calm arrangements of cells that shared a unified function, creating the first coherent layers and coordinated structures within a single organism. This quiet step, the organization of cells into tissues, marked a profound shift in evolution. It allowed multicellular bodies to move beyond mere clusters and become living systems with distinct regions, each with its own gentle purpose. To imagine these early tissues, picture a simple multicellular organism floating or anchored in ancient seas. At first glance, it might resemble a soft sheet, a hollow sphere, or a branching filament, but within this form, they were beginning to coordinate their roles, responding to chemical messages, environmental cues, and the positions they held within the structure. The outer layer of cells often faced the world directly bathed in light, exposed to gentle currents, and in contact with dissolved nutrients. These cells gradually adapted to perform duties that suited this position, becoming more efficient at absorbing light or gathering minerals. Their tasks supported the entire organism, just as the protective outer tissues of modern life support deeper layers. Inside, the cells were shielded from direct contact with the environment. Their role, therefore, became different. Some specialized in reproduction, gently dividing to maintain the structure and continuity of the organism. Others stored nutrients or reinforced internal stability. Even though each cell still carried the same essential blueprint, differences in exposure, chemical gradients, and communication began guiding them into separate roles. These internal cells formed tissues not because they were forced into these roles, but because the environment quietly encouraged them to adopt stable, consistent patterns of function. In the earliest tissues, one of the simplest and most important arrangements involved the formation of an epithelial-like layer. This layer acted as a boundary, separating the organism from the world around it. The cells in this layer adhered closely to one another, creating smooth sheets that helped the organism maintain its shape. They also regulated the exchange of substances, allowing nutrients to enter and waste to leave in ways that protected the inner regions. This basic tissue was the ancestor of all later protective linings found in plants, animals, and fungi. Though exceedingly simple by modern standards, it represented a major step toward organized structure. Other tissues formed as inner cells began to coordinate around shared tasks. In some organisms, cells that produced adhesive substances gathered in specific regions, forming supportive frameworks that helped the organism maintain its integrity. In others, cells with higher metabolic activity created zones dedicated to energy production, feeding the quieter cells deeper within. These early internal tissues were not rigid or strictly separated, but they displayed a clear shift toward differentiated roles. The organism as a whole began to behave in a more balanced, unified way. One of the earliest models of tissue organization can be observed in modern sponges, which retain structures reminiscent of ancient multicellular designs. Though sponges evolved later than the organisms we are discussing, their bodies provide insight into how early tissues may have functioned. Their outer layer consists of protective cells, while inner cells specialize in filtering nutrients or producing structural fibers. Even without true organs, they have a cooperative organization that reflects early steps in tissue evolution. Observing such modern examples allows us to imagine how early ancestors might have lived in their quiet oceans, relying on simple but effective divisions of labor. Another gentle innovation in early tissues was the development of primitive contractile cells, ancestors of what would later become muscle. In some early multicellular organisms, certain cells developed the ability to shorten slightly in response to chemical or mechanical cues. Though these contractions were weak and limited, they allowed the organism to adjust its shape, open or close simple cavities, or gently reposition itself in the water. These early contractile tissues did not produce coordinated movement but marked the beginning of physical responsiveness within the multicellular body. This soft responsiveness allowed the organism to explore its environment a little more actively, even if only on a very small scale. Tissues also developed ways to support the movement of nutrients through the organism. Since not all cells had direct access to the surrounding water, internal tissues began to form channels or porous structures that allowed fluids to flow through the body. These structures ensured that all cells, even those deep inside, received nourishment. They also helped remove waste, maintaining internal balance. This early internal transport, though simple, laid the groundwork for later circulatory systems in more complex organisms. Perhaps one of the most important features of early tissues was their ability to repair themselves. When portions of an organism were damaged, perhaps by shifting sediments, grazing microbes, or the gentle abrasion of currents, cells within the tissues could reproduce or reorganize to fill the gaps. This regenerative ability was essential in the fragile early seas, where stability depended on the quiet cooperation of many cells. Even today, some simple multicellular organisms retain extraordinary healing abilities, echoes of these ancient strategies that ensured survival without the need for complex immune or repair systems. Chemical communication played an increasingly important role as tissues evolved. Cells began sending signals not just to nearby neighbors, but sometimes across the entire organism. These signals coordinated growth, division, and responses to environmental change. In this way, tissues functioned as unified systems, with cells working together through soft, continuous communication. The beginnings of hormonal or signal-like molecules may have appeared in this stage, not yet sophisticated but adequate to guide the slow rhythms of early life. Over time, patterns of tissue arrangement became more consistent across generations. The shape of the organism, the thickness of its layers, and the placement of its interior spaces became stable traits. These traits were gently passed from parent to offspring, encouraging greater refinement through natural selection. Organisms with well-arranged tissues found better stability, more efficient ways of gathering energy, and improved methods of surviving environmental fluctuations. These advantages might seem subtle, but they were enough to guide evolution toward more intricate multicellular designs. In these early oceans, life was still simple and quiet, but the groundwork for complexity was unmistakably taking shape. Tissues represented an important transition, a move from collections of similar cells toward organized systems with distinct roles. These systems allowed organisms not just to survive, but to explore broader ecological possibilities, living in new environments and interacting with the world in new ways. As we leave this part of the story, we carry with us the gentle understanding that the formation of tissues was not an abrupt leap, but the natural outcome of millions of years of cells learning to communicate, share tasks, and shape themselves into harmonious structures. In the next part, we will drift into the world of the Ediacaran seas, where these early tissues blossomed into the first softly shaped creatures, the calm ancestors of animals as we know them. 15 gentle creatures of the Ediacaran era. In the long, unhurried timeline of Earth's history, there is a particularly soft and mysterious interval that bridges the world of simple multicellular tissues and the later flourishing of animals with distinct organs and movement. This period is known as the Ediacaran era, spanning roughly from 635 to 541 million years ago. It was a time when the seas were calm, when nutrients moved in slow cycles, and when the first large, softly shaped organisms drifted, rested, or anchored themselves across the ocean floor. These beings, neither plants nor animals in the modern sense, represent some of the earliest attempts by life to form bodies of substantial size and complexity. Their forms were strange, peaceful, and almost dreamlike, as though shaped by gentle currents rather than by the vigorous demands that later evolution would bring. To picture the Ediacaran world, imagine seafloors covered in fine sediments, illuminated by soft light filtering through shallow water. Oxygen was becoming more available. The oceans were stabilizing after ancient ice ages, and the slow accumulation of nutrients supported more elaborate forms of multicellular life. Against this serene backdrop, the first large organisms appeared. These creatures were often flat, frond-like, disc-shaped, or quilted. Their bodies lacked the rigid skeletons, muscles, or nervous systems we now associate with animals. Instead, they possessed simple tissues arranged in repetitive patterns that seemed to echo the principles of early multicellular organization described in earlier parts of this lecture. One of the most iconic Ediacaran organisms was Dickinsonia, a softly ribbed, oval-shaped being that could reach nearly a meter in length. It had no known mouth, gut, or limbs. Instead, its thin, quilt-like body may have absorbed nutrients directly from the microbial mats covering the ocean floor. Its shape was calm and symmetrical, with segments arranged in gentle arcs that radiated outward from a central line. The simplicity of its form suggests that it relied on diffusion and surface contact rather than internal organs to sustain itself. Yet its size indicates that it successfully navigated the environmental opportunities of its era, thriving for millions of years in these peaceful seas. Another common Ediacaran form was Fractophusis, a branching organism whose frond-like shape spread across the seafloor like a delicate pattern traced by water itself. The repeats within its structure, small branches forming larger branches, reflect fractal organization, a natural pattern that arises when similar processes occur at multiple scales. This recurring pattern suggests that early multicellular forms were still relying heavily on environmental gradients and simple developmental rules to shape their bodies. Fractophusis may have reproduced by releasing small propagules that drifted in the water before settling and growing into new fronds. This quiet method of reproduction allowed it to form clusters across the seafloor, creating some of the earliest structured communities of large organisms. Many Ediacaran organisms were anchored to the seafloor, gently swaying in the currents. Charniodiscus, for example, grew on a stalk that supported a leaf-like frond. This upright posture allowed it to capture nutrients or sunlight more effectively than flat organisms. Its shape reveals a new level of structural organization, tissues dedicated to attachment, others designed for exposure to the environment, and a body that maintained its shape even as water flowed past. Charniodiscus and other frond-like forms represent an early step toward the three-dimensional body plans that would later appear in more complex animals. Still others, such as Sprigina, hinted at even greater complexity. Sprigina possessed a head-like end and a gently segmented body, features that seem to foreshadow the bilateral symmetry common in later animals. While it lacked bones, organs, or a defined nervous system, its shape implies the beginning of directional movement and a simple differentiation between the front and back of the body. This kind of organization likely emerged from the continued refinement of tissues and communication systems, allowing cells to adopt new roles depending on their position within the body. Despite this growing diversity, Ediacaran organisms shared important characteristics. Most had soft bodies composed of flexible tissues arranged in repetitive, symmetrical patterns. They were likely slow-moving or stationary, relying on diffusion, gentle water flow, and simple feeding strategies. Many lived in close association with microbial mats, thin layers of bacteria and algae that blanketed large portions of the seafloor. These mats provided food, stability, and even shaping influence, as some organisms grew directly on their surfaces, leaving behind faint impressions that became preserved in ancient sediments. The Ediacaran landscape was unlike anything that exists today. There were no predators as we would understand them, no animals burrowing deeply into the sediment, and no organisms with complex digestive systems or skeletons. This absence of ecological pressure allowed these early creatures to flourish in peace. Their forms were shaped more by the environment than by competition. The calm waters and stable sediment fields of the Ediacaran seas supported life that expanded slowly, spreading across the seafloor like living gardens. The emergence of these organisms also reflects the culmination of earlier evolutionary steps. The ability to form stable tissues, respond to environmental gradients, and distribute nutrients across a large body allowed these creatures to grow in ways no previous life form could. Their existence shows how life was gently experimenting with new possibilities, forming bodies that were larger, more patterned, and more refined than anything before. They represent a quiet threshold, life stepping softly into complexity while still remaining rooted in simple principles of organization. Yet even within this gentle world, subtle changes were preparing the way for future transformations. As some Ediacaran organisms developed slightly thicker tissues or primitive internal spaces, they began exploring new ways of maintaining stability. As others developed faint directional structures, they opened the possibility of movement and interaction. These soft innovations laid the foundation for the increased complexity that would come during the Cambrian period, when animals with more defined organs and behaviors would appear. But in the Ediacaran era, life remained peaceful and largely static. The seas were filled with delicately patterned bodies, resting in stillness. Their existence reminds us that complexity does not always arise through struggle. Sometimes it grows through calm exploration, as cells and tissues learn to arrange themselves into forms that reflect both their inner cooperation and the gentle conditions of their environment. As we prepare to move forward into the next part, we will step into the early stages of true animal evolution, still soft, still slow, but beginning to show the first signs of systems that will one day give rise to fully formed creatures. 16. The first simple animals take shape. In the slow and tranquil unfolding of Earth's early history, after the quiet gardens of the Ediacaran seas had settled into place, a new kind of being began to emerge. These beings were still soft and simple, still connected to the ancient rhythms of water and light, but they possessed something that set them gently apart from their predecessors. They were true animals, organisms composed of different types of tissues working together in quiet coordination. Their bodies were small and delicate, lacking bones or shells, but within them lay the earliest hints of systems that would one day support movement, sensation, and purposeful interaction with the world. These first simple animals did not disrupt the calm oceans. Instead, they added new layers of subtle complexity to the serene underwater landscapes. To imagine these early animals, we must picture organisms that were neither fully modern nor entirely primitive. They did not have eyes, limbs, or organs as we understand them today. Yet they had developed tissues that allowed water to flow through their bodies, carrying nourishment to every cell. These gentle creatures lived in shallow seas where light filtered softly from above and microbial mats still carpeted large portions of the seafloor. They depended on the constant movement of water for survival, relying on the quiet interplay between their bodies and the surrounding environment. One of the earliest and simplest examples of true animal life can be seen in organisms resembling modern sponges. Sponges represent one of the oldest surviving animal groups, and their bodies offer insight into what the first animals might have been like. They are composed primarily of two layers of simple tissues with a jelly-like substance between them. These tissues contain cells that specialize in filtering water, drawing it through small openings and extracting nutrients along the way. The structure is so gentle and efficient that it requires no movement on the part of the organism. Water, guided by the sponge's porous body, flows naturally through internal channels, bringing nourishment and carrying away waste. In these early animals, the quiet choreography of water and tissue replaced the need for complex organs. The simplicity of these early animals did not limit their effectiveness. Their porous bodies provided a large surface area for absorption, and their tissues were arranged in patterns that supported stability and steady contact with the environment. Some were anchored to rocks or sediments, while others floated freely with slow currents. Their forms varied naturally. Some were branching, others tubular, and still others formed mats or rounded structures. Yet each shape represented a gentle solution to the same challenge: how to bring nourishment into the body without moving or feeding actively. This quiet adaptation allowed these organisms to thrive for millions of years in peaceful marine environments. Another early group of simple animals included organisms resembling modern cnidarians, the lineage that now includes jellyfish, corals, and sea anemones. The ancestors of this group appeared in the late Ediacaran and early Cambrian periods, though their earliest forms were likely even simpler than their modern descendants. These organisms possessed tissues arranged around a central cavity, a primitive digestive chamber that allowed them to ingest food rather than absorb it directly through the body surface. This cavity marked an important step in animal evolution: the internalization of digestion. Instead of relying solely on passive absorption, early cnidarian-like organisms could trap small particles or absorb dissolved nutrients within a protected interior space. Their bodies often took on radial symmetry shapes that were organized around a central point, like spokes around a wheel. This symmetry suited their still and drifting lifestyles, allowing them to experience their surroundings evenly from all directions. The tissues that formed their simple bodies were arranged in layers: an outer layer for protection and sensing, an inner layer for digestion, and a gelatinous middle layer for support. These layers did not yet form organs, but they demonstrated a level of organization far beyond that of earlier multicellular life. Even without a brain or nervous system, these organisms could respond to touch or chemical cues through simple networks of cells, marking the earliest beginnings of animal sensation. In some regions of the ancient oceans, early animals formed communities on the seafloor, where they lived among the soft-bodied organisms of the Ediacaran era. Their presence subtly changed the environment. By filtering water, they affected nutrient availability. By growing on rocks or other hard surfaces, they created new habitats. These interactions were gentle but meaningful. The early animals were not predators. They did not disturb the world through force or movement. Instead, they influenced their surroundings through steady ecological presence, becoming part of the quiet web of interactions that shaped marine ecosystems. As these early animals grew more common, new patterns of body organization began to appear. Some organisms developed tissues capable of slight, rhythmic contractions. These contraction-capable cells were the ancestors of muscle tissues. Their early movements were simple, perhaps closing a cavity, curling a body edge, or helping water move through internal spaces. Yet these subtle motions gave early animals a small measure of control over their environment. They could adjust their bodies to currents, alter the shape of their cavities, or reposition themselves slightly on the seafloor. These tiny adjustments represented significant evolutionary steps. For the first time in life's history, organisms were beginning to influence their surroundings through gentle motion. Near the same time, other early tissues formed primitive nervous systems. These were not centralized networks or brains, but distributed webs of cells capable of sending electrical signals. These signals allowed organisms to coordinate contractions, respond to changes in light or chemicals, and interact with the environment in new ways. The earliest nervous tissues helped guide the flow of water through the organism, regulate feeding structures, and coordinate simple reactions. These soft exchanges of information were the first sparks of sensory awareness in animals. Some early animals developed tube-like bodies with openings at either end, one for bringing in water or food, and another for expelling waste. This one-way flow represented another gentle but important evolutionary innovation. It allowed these organisms to process food more efficiently and maintain internal stability. The development of tubes and chambers within these bodies reflected a growing trend. Life was learning to create spaces within itself, to fold its own tissues in ways that enhance survival and function. As these early animals spread through ancient seas, they coexisted with Ediacaran organisms, microbial mats, and other simple multicellular forms. The world remained quiet, its ecosystems still dominated by slow growth, gentle interactions, and soft-bodied creatures. Yet life was changing. The emergence of tissues capable of coordination, movement, and communication set the stage for greater complexity. These early animals were like softly spoken words in a language that evolution was just beginning to form, a language that would later express itself in the intricate forms of the Cambrian explosion. And yet, despite their importance, the early animals of this era remained modest. They did not rush or dominate. Instead, they represented balance, bodies shaped by tissues that had learned to cooperate deeply, to build spaces, to move gently, and to sense the world in its calmest form. They lived their lives in harmony with the slow rhythm of ancient seas, revealing how multicellular existence could evolve from simple patterns into structures with both function and quiet beauty. As we move into the next part, we will explore how other multicellular lineages, namely fungi and plants, began their own gentle journeys, branching from early eukaryotic ancestors and adding new dimensions to Earth's slowly diversifying life. 17 fungi and plants begin their ancient journeys. In the slow, unfolding story of early multicellular life, while simple animals were learning to filter water and coordinate the first soft contractions, two other great lineages were quietly beginning their own long journeys, the fungi and the plants. Their origins stretch back to the same ancient eukaryotic ancestors that gave rise to animals. Yet their destinies grew in different directions, shaped by the calm interactions between cells, water, minerals, and sunlight. These lineages did not appear suddenly as we recognize them today. Instead, they emerged gently, step by step, through simple associations and soft collaborations between cells and their environments. Their early forms were subtle and often microscopic, but they carried the seeds of the vast diversity that would one day coat the Earth's landscapes with forests, mosses, lichens, and hidden fungal networks beneath soil and stone. To imagine the beginnings of plants and fungi, we must return to the quiet oceans where photosynthetic cells were thriving in sunlight-rich shallows. These photosynthetic ancestors, likely early algae, held within their cells structures descended from ancient endosymbiotic events. Through calm cooperation, early eukaryotes had taken in cyanobacteria, which gradually became chloroplasts, the organelles responsible for gathering sunlight and nourishing the cell. These algae existed as simple sheets, filaments, or spheres, absorbing the gentle warmth of the sun and drifting with soft currents. They depended on the same simple patterns that had shaped early multicellular life: gradients of light, flows of nutrients, and the whisper-like communication between neighboring cells. In time, some of these algae began forming more stable colonies. Outer cells became more efficient at capturing light, while inner cells contributed structural or reproductive roles. This gradual patterning laid the initial foundation for what would someday become plant tissues. However, these early forms remained entirely aquatic. Their bodies were soft and flexible, adapted to life suspended in water. They required no support structures, no vessels, and no protective coatings. Alongside these emerging algae, another lineage was taking shape, one that did not rely on sunlight at all. Early fungi evolved from unicellular ancestors that consumed organic matter rather than producing their own food. These primitive fungi lived in shaded environments on the ocean floor, on decaying algae, or in close association with bacteria. They absorbed nutrients directly through their cell walls, using enzymes to break down complex molecules into simpler forms. This feeding strategy made fungi profoundly different from early plants, yet both lineages shared a deep reliance on cooperation plants through the union with photosynthetic symbionts, and fungi through the steady breakdown and recycling of organic material. Early fungi were likely small and thread-like, forming delicate networks known as hyphae. These hyphae spread through soft sediments or across decaying matter, extending their reach gradually, cell by cell. As they grew, they formed intricate branching patterns that increased their surface area, allowing them to absorb nutrients more effectively. These networks did not move quickly. They expanded at a gentle pace, settling into the stillness of their environment. Their quiet role was essential. By breaking down material that other organisms could not use, they opened new pathways for nutrient cycling in early ecosystems. The presence of both algae and fungal ancestors created opportunities for interaction. In some places, early fungi began forming associations with photosynthetic organisms, wrapping their soft hyphae around algal cells or embedding themselves within algal tissues. These early interactions were the faint beginnings of symbioses that would one day give rise to lichens, remarkable partnerships that enabled life to flourish in some of the most challenging environments on Earth. In these early oceans, however, such relationships remained simple, offering mutual benefits. Algae received minerals from fungi, and fungi received sugars produced by photosynthesis. These early partnerships demonstrated how cooperation between distinct lineages could create biological stability even before life ventured onto land. Meanwhile, the algae themselves were growing more varied. Some remained microscopic, drifting freely in currents. Others formed thicker shields of tissue that helped them withstand stronger water movements. A few began to anchor themselves more firmly to submerged rocks, developing holdfasts that resembled early roots. These holdfasts did not absorb nutrients at first. Instead, they provided stability, allowing the algae to grow upward toward sunlight. This simple anchoring behavior represented one of the earliest steps toward the concept of a body that interacts with its environment in distinct, specialized ways. In deeper and more shaded waters, certain algae evolved pigments that allowed them to capture sunlight at wavelengths unavailable to their green relatives. These changes increased the depth range where photosynthesis could occur, gently expanding the ecological niches available for plant-like organisms. Over time, different groups of algae diversified into red, brown, and green lineages, each adapted to particular light and nutrient conditions. These variations illustrated how life responded subtly to shifting environmental patterns. Although not yet true plants, these algae laid the biological groundwork for the vast terrestrial flora that would eventually shape the planet. The early development of fungi continued in parallel. By breaking down dead organisms, both unicellular and multicellular, they provided essential minerals to the surrounding waters. This slow, steady transformation of organic material helped stabilize ecosystems, ensuring that nutrients remained available even as life grew more abundant. Without fungi, the earliest ecosystems might have stagnated under the weight of their own remains. Instead, fungal activity kept the oceans gently renewed, supporting the continued emergence of larger and more diverse life forms. While these early interactions unfolded, the world itself was changing. Continents were shifting, shallow seas expanded and contracted, and the atmosphere continued to accumulate oxygen. These environmental shifts influenced the distribution and evolution of early algae and fungi, encouraging some lineages to explore new habitats. In sheltered tide pools or along gently sloping shorelines, some algae may have experienced brief exposures to air as the tides receded. These moments, quiet, rhythmic, and recurring, created subtle pressures that encouraged algae to tolerate short periods of dryness. Over countless generations, these early adaptations would evolve into the traits necessary for life to eventually move onto land. Fungi, with their ability to absorb moisture from surfaces and break down minerals, were equally well equipped to explore emerging coastal environments. Their hyphae could spread across damp surfaces, anchoring themselves and absorbing nutrients from exposed sediments or decaying organic matter. In time, these quiet explorations beyond the water's edge formed the earliest biological footprints on land. Soft, hidden steps, taken long before true plants or animals would follow. Together, these two lineages, algae evolving toward plants, fungi evolving toward their vast underground networks, represent two complementary paths of early multicellular evolution. Plants would one day bring oxygen, food, and structure to terrestrial ecosystems. Fungi would become partners, recyclers, and stabilizers, weaving themselves through soil, stone, and root. Yet in these early oceans, their beginnings were humble, shaped by simple interactions, slow adaptation, and the gentle guidance of environmental gradients. As we move forward from this part, we will follow the deepening branches of life's tree, watching as multicellularity radiates outward into an ever-widening array of forms. The next chapter brings us into the story of how life diversified further, how countless lineages, from algae to animals, began splitting into new paths in the great branching tapestry of evolution. 18. The great branching of life's tree. As the oceans of early Earth continued their calm transformations, the living world entered a period where the diversity of multicellular forms began to spread outward in gentle but distinct directions. This expansion did not happen with sudden leaps or dramatic bursts of novelty. Instead, it unfolded slowly, like the branching of a soft coral or the spreading of roots beneath quiet soil. Each lineage, whether plant-like, fungus-like, or animal-like, followed its own path, guided by subtle pressures and opportunities offered by the environment. Together, these paths formed the earliest branches of what we now call the tree of life, a vast, interconnected structure where every species traces its origin back to simpler ancestors that lived in the stillness of ancient seas. To picture this branching, imagine a single trunk representing early eukaryotic cells, organisms that had achieved internal complexity with nuclei, organelles, and the capacity for cooperative interactions among their parts. From this trunk emerged several major lineages, each exploring its own possibilities for survival. Some remained unicellular, drifting freely through the oceans and thriving in their simplicity. But others ventured into multicellularity, forming tissues, colonies, and eventually bodies capable of performing multiple functions. As these lineages explored new forms, they created patterns that became the basis for the immense diversity of life that exists today. The branching of life was not linear. It was a gentle, rhythmic unfolding, with each lineage responding to its own set of ecological pressures. Early plants descended from photosynthetic algae, diversified into shapes adapted for different levels of light and nutrient availability. Some grew in shallow, sunlit waters, forming broad, leaf-like tissues that captured light efficiently. Others found homes in deeper waters, developing pigments that allowed them to absorb wavelengths of light that penetrated further into the sea. These quiet shifts in pigment and structure represented the early diversification of plant life, which would one day dominate the land with towering forests and intricate root systems. Fungi, following their own subtle trajectory, branched into lineages that specialized in decomposing various types of organic matter. Some became adept at breaking down tough structures like cyanobacterial mats, while others formed delicate partnerships with algae or bacteria. These early symbioses laid the groundwork for the remarkable relationships fungi maintain today with nearly every plant lineage. Through these associations, fungi and early plant-like organisms explored new ecological niches, gradually increasing the complexity of life's interactions. This cooperative branching helped stabilize early ecosystems and ensured the recycling of nutrients across vast stretches of time. Animal lineages also began branching in their own quiet ways. The earliest true animals, such as sponge-like organisms, remained simple but diversified into different shapes and sizes, suited to various water flows and nutrient concentrations. Some developed branching bodies to increase their filtering efficiency, while others grew into more compact forms that resisted the movement of currents. Although their tissues remained primitive, these organisms laid the foundation for future animal diversity by experimenting with body architectures that allowed for subtle differences in function. Among the most intriguing branches were those that eventually gave rise to organisms with rudimentary nerve nets and contractile tissues. These early developments allowed organisms to respond to stimuli in new ways, even if their reactions were slow or limited. As these creatures diversified, they created early versions of ecological interactions such as gently grazing microbial mats, adjusting their posture to currents, or opening and closing simple cavities. Though these activities were subtle, they helped shape the marine environment and encouraged further diversification of both plants and animals. The branching of life during this period was deeply influenced by the availability of nutrients, oxygenation of the oceans. As oxygen accumulated gradually in the atmosphere and dissolved into the waters, some organisms evolved to use it more efficiently. Oxygen, while gentle in presence, allowed for more energy to be extracted from food and sunlight, supporting the evolution of larger and more active bodies. Organisms that could tolerate or utilize oxygen found new opportunities for growth, while those that preferred anoxic conditions retreated into deeper or more secluded areas. This separation gently guided lineages toward specializations that would later appear as distinct ecological roles. Communication between cells also played a vital role in branching. As organisms grew larger and more complex, they required new ways to coordinate their internal activities. Chemical gradients, signal molecules, and early pathways of genetic regulation began shaping how cells behaved during development. These regulatory patterns were passed down to offspring, becoming the earliest forms of what we now recognize as developmental programs. Over time, the repeated use of these programs produced consistent body patterns within each lineage, deepening the distinctions between groups and helping to define their evolutionary paths. As each lineage explored its own possibilities, interactions between the lineages also increased. Early plant-like organisms changed the composition of the water by releasing oxygen and organic matter, which influenced fungi and animals. Fungi, through decomposition, released minerals that fed algae and early plants. Animals, by filtering water or stirring sediments, created small changes in flow patterns and nutrient availability. These interactions created mild but meaningful pressures that pushed each lineage to continue diversifying. Over long periods, the peaceful interplay between organisms guided the branches of life toward greater complexity. The gradual branching of life also created ecological layers. Some organisms lived on the seafloor, forming stable communities of sessile forms. Others floated freely, drifting in the sunlight near the ocean's surface. Some explored shaded crevices, while others occupied nutrient-rich zones near upwellings. This layering of habitats encouraged lineages to adapt to different conditions, each discovering its own niche. The diversity of environments available in ancient oceans helped drive the spreading branches of the tree of life, encouraging subtle shifts in shape, behavior, and structure. As diversity grew, so did the stability of ecosystems. When one lineage expanded, others often found ways to make quiet use of the new conditions. When one group began filtering water more efficiently, others adjusted by growing on their surfaces or in their shadows. This balance meant that expansion did not lead to disruption, but rather to new opportunities for coexistence. Over millions of years, these interactions formed the basis for complex ecosystems, long before mobile animals and structured plants dominated the earth. Throughout this branching, the deep unity of life remained. All lineages retained the same basic cellular structures inherited from early eukaryotes. They maintained the same principles of reproduction, growth, and adaptation. And despite their differences, they shared the quiet foundation described in cell theory, that every living organism arises from pre-existing cells, and that these cells carry the genetic information needed to guide life forward. As we reflect on this great branching, we see that it was not marked by competition or conflict, but by exploration life, gently testing new ways of existing, new shapes, new cooperative strategies, and new ecological roles. Each branch grew outward, creating variations that would one day culminate in the incredible tapestry of life on Earth. In the next part, we will explore how this branching continued into the present, and how multicellularity remains an ongoing story, still unfolding, still adapting, and still discovering new forms in the quiet corners of the natural world. 19. Multicellularity as an ongoing story. In the long, gentle arc of life's history, multicellularity is often spoken of as though it were a single event, one moment of transition when solitary cells first decided, in their quiet, natural way, to remain together. But in truth, multicellularity is not a chapter that ended millions of years ago. It is an ongoing story, one that continues to unfold in the oceans, in the soil, within plants and fungi, and even inside organisms whose lineages stretch back to those earliest experiments. Evolution never stopped refining, reinventing, and rediscovering the benefits of living as many cells bound in simple cooperation, in small ponds, deep seas, forming new versions of multicellular existence that echo ancient beginnings while revealing entirely new forms. To understand multicellularity as a continuing process, it helps to return to the principle that guided its earliest steps. Cells discovered that collaboration could make life more stable. As environments shifted, as nutrients cycled in slow rhythms, and as oxygen levels rose and fell through the ages, countless lineages found themselves in situations where remaining together offered an advantage. This advantage was never abrupt. It was an accumulation of small benefits: protection from currents, improved nutrient access, shared metabolic tasks, and the ability to form environments that buffered each cell from the changing world outside. Even today, these quiet benefits continue to shape how new forms of multicellular organization arise. Modern algae offer striking examples of how transitional multicellularity remains active. Some species live freely as single cells for most of their existence, dispersing effortlessly through water. Yet when environmental conditions shift, perhaps when light becomes scarce or nutrients diminish, they gather into clusters, forming colonies that behave as unified organisms. These colonies may have just a handful of cells, or they may contain hundreds, arranged in simple spheres or sheets. When conditions improve, the colonies may disperse again, returning to individual existence. This flexibility mirrors the earliest, most experimental stages of multicellularity, when life was still learning what it meant to form stable bodies. These algae remind us that multicellularity is not a fixed state but a fluid continuum between togetherness and individuality. In microbial communities, the story is similar. Bacteria often form biofilms, thin, coordinated layers that adhere to surfaces and create shared environments. These biofilms consist of millions of individuals, each contributing to a collective structure that offers protection and stability. Though bacteria remain unicellular, their cooperative behavior echoes the principles of multicellular life: division of labor, communication through chemical signals, and shared benefit. Some bacteria even create channels within their biofilms, allowing nutrients and waste to move through the community much as they would in simple tissues. These modern microbial communities demonstrate that multicellularity-like organization can arise repeatedly whenever cooperation offers survival advantages. Life, it seems, is always exploring new ways to collaborate. Even in more complex organisms, multicellularity continues to take unexpected forms. Some fungi remain primarily unicellular but can switch into multicellular growth patterns when conditions favor spreading through soil or decaying wood. Their hyphae, long, thread-like structures form vast networks that function almost like underground organs, absorbing water, redistributing nutrients, and interacting with plant roots in subtle exchanges. These fungal networks blur the line between individual and collective life, much as early multicellular organisms once did. They show that even within lineages deeply committed to multicellular existence, plants, too, continue to refine and reinvent their multicellular strategies. In mosses and liverworts, the earliest plant lineages to step onto land, we see simple tissues forming mats and soft leafy structures that thrive in moisture-rich habitats. Their cells communicate through gentle chemical gradients, just as algae once did. Higher plants later evolved more complex tissues, yet even today they maintain the quiet principles of early multicellularity: coordinated growth, division of labor between tissues, and the ability to respond as a whole organism to changes in light, water, and soil. In their seeds, spores, and root tips, plants revisit early developmental patterns with each new generation, building multicellular forms from the same soft beginnings. Animals, despite their complexity, also reveal how multicellularity remains dynamic. Regeneration offers one of the clearest examples. Some animals, such as sponges, hydra, and certain worms, can reorganize their cells into whole new individuals if their bodies are disrupted. This remarkable ability reflects ancient developmental rules that never fully departed from these lineages. In a sense, such creatures remind us of the earliest multicellular forms, flexible, responsive, and guided by simple interactions between cells. Even in the most advanced animals, from insects to mammals, the ongoing story of multicellularity unfolds at the microscopic level. Each new organism begins as a single cell, which then divides, communicates, and organizes into tissues following principles inherited from ancient ancestors. While the resulting forms are far more intricate than those of early multicellular organisms, the underlying processes remain grounded in gradients, signals, and cooperation, the same tools that shaped life's earliest tissues in quiet oceans long ago. The persistence of multicellularity as a living process also becomes clear when we observe social organisms. Although not multicellular in the strict biological sense, colonies of ants, bees, and termites behave like superorganisms, complex groups acting as a single cohesive unit. Members take on specialized roles, communicate constantly, and maintain the stability of the colony through coordinated behavior. These societies reflect the same principles that shaped early multicellular life: cooperation, communication, and the sharing of resources. They demonstrate that the benefits of collective living extend well beyond the boundaries of individual cells. The ongoing evolution of multicellularity also appears in scientific observations of laboratory organisms. Under certain conditions, unicellular species can evolve multicellular traits in just a few hundred generations, forming clusters, developing early specialization, and evolving adhesion molecules that allow them to stick together. These experiments highlight how pervasive and natural the transition to multicellularity is when conditions favor cooperation. They show that the emergence of multicellularity is not an ancient event locked in the distant past, but a potential outcome that still lies within the reach of life whenever circumstances create the right balance of opportunity and challenge. The story of multicellularity continues not only in natural environments but in the very structure of ecosystems. Forests, coral reefs, and tide pools all depend on interactions between multicellular organisms and their surrounding communities. Mycorrhizal fungi weave through forests, linking the roots of trees in nutrient-sharing networks. Corals form partnerships with algae, building vast ecosystems filled with countless forms of life. These systems are so interconnected that they function almost like extended multicellular bodies, each organism supporting the others through subtle, continuous exchanges. As we reflect on multicellularity's ongoing journey, we see that it is not a fixed or final state. It is a way of living that life continually revisits, refines, and reinterprets. From simple colonies of algae to the complex bodies of plants and animals, from microbial biofilms to intricate ecosystems, multicellularity is a gentle thread weaving through all of evolution. It reminds us that cooperation, communication, and collective stability are not only ancient achievements, but living processes that continue to shape the world around us. In the final part of this lecture, we will return to the deepest theme of all, the unity of living things, and how every multicellular organism, including ourselves, traces its origins to the first cells that chose, in quiet companionship, to face the world together. 20. A soothing closing. The continuity of all living things. As we come to the end of this gentle journey through the origins of multicellular life, it becomes clear that the story we have been exploring is not simply a sequence of ancient events, but a soft and continuous thread that binds every living thing together. From the simplest clusters of cells drifting in ancient waters to the rich diversity of plants, fungi, and animals that now inhabit forests, oceans, and quiet ponds, life has always been guided by the quiet companionship of cells choosing to remain near one another. This companionship, rooted in cooperation and shaped by countless environmental rhythms, created the foundation upon which all complex life now rests. To appreciate this continuity, imagine looking across the long arc of Earth's history. In the earliest chapters, single-celled organisms filled the oceans, responding only to light, temperature, and the gentle pull of currents. Their lives were solitary, yet the oceans were dense with possibility. When some cells began to cling together first loosely, then with growing stability, an entirely new dimension of life opened. These clusters, held by simple bonds and shaped by environmental gradients, became softly unified bodies, drifting in the sunlit shallows. With each passing generation, they learned new ways of communicating, coordinating, and organizing their inner worlds. These changes were subtle but steady, like the slow smoothing of a pebble beneath waves. As multicellularity matured, life discovered harmony through differentiation. Some cells absorbed light, others stored nutrients, and still others reproduced or supported structure. These early roles were gentle beginnings of what would become tissues. Over millions of years, the seas filled with forms that hinted at the future. Soft fronds, quilted bodies, branching filaments, and the first simple animals. All were quiet expressions of a deeper truth that cooperation could lead to stability, and stability could lead to complexity. This pattern did not end with the ediacaran creatures resting peacefully on ancient seafloors. It continued with the ancient fungi, spreading their delicate threads through sediments, and with early algae, lifting themselves toward light with gentle determination. It continued as plants learned to anchor themselves, to harness sunlight even in shallow tidal pools, and eventually to step softly onto land. Every new lineage, whether plant, animal, or fungus, carried within it the echoes of that earliest decision, the quiet choice of cells to face the world together. Even now, in the modern world, we see reflections of these ancient beginnings everywhere we look. In the tissues of plants, where layers of cells coordinate growth through sunlight and water, in the delicate gills of mushrooms, where hyphae spread in intricate patterns reminiscent of early multicellular networks, in the bodies of animals, where trillions of cells cooperate seamlessly through signaling pathways that have roots in early chemical communication. Whether in the branching of coral reefs, the filaments of algae, or the soft biofilms resting on stones in streams, the principles of multicellularity remain alive and present. The continuity becomes even more profound when we consider how each multicellular organism begins its own life. Every plant sprout, every fungal spore, every juvenile animal begins as a single cell, much like the solitary ancestors from which multicellularity first emerged. This single cell divides and organizes itself according to ancient rules shaped by billions of years of evolutionary experimentation. The gradients, signals, and cooperative patterns that guided early clusters still guide development today. In the unfolding of each new organism, the story of multicellularity is retold quietly, generation after generation. Even at the level of ecosystems, we see echoes of multicellularity's earliest lessons. Forests behave like vast interconnected organisms, with roots, fungi, and soil forming a network through which water and nutrients pass. Coral reefs build intricate structures that shelter countless species, much like early microbial mats supported emerging multicellular forms. Even human societies, though vastly more complex, rely on cooperation, communication, and shared purpose principles that have guided life since its earliest steps toward collective existence. This continuity is soothing because it reminds us that life is not chaotic or abrupt. It unfolds slowly, in gentle rhythms, shaped by the same patient forces that guided the very first cells: light, nutrients, temperature, communication, and simple structural patterns. These quiet elements have been life's companions across deep time. They have shaped not just the bodies of organisms, but the entire character of living systems. When we reflect on the full journey of multicellularity from its humble origins to its ongoing expressions, we find a story that is both ancient and deeply present. We see that complexity did not arise through conflict, but through cooperation. Diversity did not emerge through sudden leaps, but through millions of soft adjustments. Life grew not by abandoning its past, but by building gently upon it, adding new layers of organization while retaining the quiet principles of its earliest moments. And in this calm, sweeping continuity, we find a reminder of our own place within the story. Every cell in our bodies carries the memory of ancient oceans, where early organisms first learned to share tasks, to communicate, and to form simple tissues. Every heartbeat, every breath, every moment of growth within us is guided by processes that trace back to those first networks of cells cooperating in the sea. Whether we are aware of it or not, we are living embodiments of the same gentle evolution that shaped the first multicellular bodies. As we bring this lecture to a close, we step away, not with finality, but with an understanding that the story continues within organisms, within ecosystems, and within every new generation that forms from a single, dividing cell. Multicellularity remains an ongoing journey, a peaceful unfolding that links all living things through time. It is a quiet reminder that life thrives not through isolation, but through connection, through the simple, enduring choice of cells to stay together, to communicate softly, and to build something greater than themselves. In that unity, the ancient and the modern meet. The oceans that nurtured early life still breathe beneath the world's surface. The principles that shaped early multicellular bodies still guide cells today. And the gentle story of evolution continues, moving forward in calm, continuous steps, just as it always has, and just as it always will.